it's your college and all? It's going
Ma'am,
good.
same as usual. Generation Z.
Mm.
What we can do?
Yeah. Yes, yes. Then,
The
ma'am,
same.
what's special?
Then nothing special, ma'am. As usual, working.
Pardon, ma'am?
Planning to come Mysuru. Let's see. Planning to
Uh,
come Mysuru.
yes, please
Waiting
come,
for
come.
some other conferences
Yeah.
also.
Yes, yes, please do come, ma'am. We can
Yes,
meet.
ma'am.
For me, 1 month rest, so I'm on rest only.
Okay, no problem for that, ma'am. Rest is also
Yeah,
needed for us.
yes, it's needed and it's good also, ma'am. If I'm in college, so I could had a lot of other works, so I could have not
Mm-hmm.
attended your conference. So
Mm-hmm.
everything went for good things only.
Yes. Sometimes we should also give time to ourselves.
Yeah, yeah, that's true, ma'am.
Just we will wait and the others to get connected.
Yes, ma'am. Let them.
Mm.
How many participants are there, ma'am?
Ma'am, 50 plus.
Oh, good.
That too also, ma'am, I don't think so. Like 50 plus is a good like ratio.
No, no,
Or a
no. Like
conference, ma'am.
when I do a phone call, I'll tell you, ma'am. Like it's
Mhm.
difficult to gather.
Yes, ma'am.
You're doing good work.
Thank you, ma'am. But still, people they don't want to learn new things. They are happy with the old things only.
Yeah, yeah.
If we try to make them
Yeah.
learn, they will say, "Oh, what to do? It's waste of
Yes,
time and
yes,
other
yes.
stuff."
But everywhere they have to make mandate, ma'am. Then only they will.
Yes, in my college, I always try to make it mandatory for all the students. Actually, whatever I'm having A&M and GNM, but then also I
Hmm.
used to say them just better you can try the new good things
Yeah,
that we
that's
can
good.
have some. We can
Yeah,
like we will explore new things, no, ma'am.
yes, ma'am. Like a new learning should happen,
Yes.
or else it will become like stagnant knowledge. So whatever they have learned years ago, so they won't know the what changes are there.
Mm. So we will,
Yeah, we'll wait for them, ma'am.
yeah, ma'am, we will wait for them, ma'am.
Yeah, okay. Hello, ma'am. Am I audible?
Yes, ma'am.
Okay, ma'am. Let me know when should I start?
Yes, ma'am. We will wait for another 5 minutes. That other person also should they can join,
Sure,
ma'am.
sure, sure,
Yeah, ma'am.
sure, ma'am. Thank you, thank you.
Yes.
Ma'am, can open the mic.
Good morning, all. Good morning, dear delegates. Today's resource person and the organizing persons. So, I welcome all the delegates for this precious and knowledgeable international conference where you are going to gain your knowledge. So, let me start the first session of today's. Let me introduce today's resource person. So, today's resource person.
Yeah, the today's resource person is Dr. Kritika Tekwani, ma'am. She is an academic associate IIM Ahmedabad. So, as her experience, she's having of 7 years of experience. As her expertise, she's having goods and service tax, income tax, tally, ERP 9, SPSS 26, that is T test, Z test, ANOVA, correlation, regression, pair T test, chi-square test, validity and reliability test, Manley U test, and she has done a PhD on goods and service tax. And even we can say that she's having several years of experience, and she has also worked in the office of joint director industries for making Rajasthan export strategy in 2020, Udyog Bhavan Jaipur Rajasthan in 2020. She has cleared NET JRF in management in 2019, and even she has cleared NET 3 times consecutively in 2019, 18, and 17. Even she has done MCOM in business administration from the University of Rajasthan in 2018. As she is also done MBA, that is in finance, from Central University of Rajasthan in 2015. She is the gold medalist in MBA. She did BCom in 2013. She got first rank in her college and 7th rank in the University of Rajasthan. So, ma'am, we would like to invite you for your speech. So, just it's over to you, ma'am.
So thank you so much for your kind words, and I hope I'm sharing my screen. If it is visible, do let me know.
Ma'am, it's not visible. If you want, I can share.
No, ma'am. I think now it will be visible. Okay, I'll just check. Is it visible, ma'am, now?
No,
Is it
ma'am.
visible?
No.
Yes, ma'am, it is visible now.
Okay, okay, fine, fine, fine. Thank you, thank you. Okay, so today I'm going to talk about the statistical tasks and the techniques, and this topic is not a theoretical topic. So I'll do the hands-on through the software, so that all the audience and all the STEAM faculties from the different institutes, they can have the some knowledge about it. I know they have the knowledge already, but it's still some updations and all things have been going on as it's a research, of course. So statistical test and the techniques is mainly used when when we have the data and we want to make some interpretation, make some output, and make some judgments, decisions. So we need some softwares, we need some tools, we need some techniques. On the basis of that, we do that, right? Okay, so firstly, we should know what kind of data you have. Like if you have a descriptive data, if you have inferential data, you know, and what is your population, what is your sample. So just to I'm just giving a brief of 2 minutes related to that, right? So if you are doing a primary data interpretation. So firstly, you have one questionnaire, you have some interview or schedule thing, so that you can get that filled from the online, also from the from the physical mode also. So like that we collect the data, right? And another thing, when the already data which is being published somewhere, like annual reports and data of financial data of any company, like their balance sheet, their profit and loss account, their cash flow statements. So that data which has already been worked by someone, and you are having that data to make some inferences from the what's going on, right? Whether simply now people are not from the commerce background, if they are from the other background also, they are also investing in some funds and all. So they are doing some analysis, see what will be the price will be hike or not. So stat is just not only be used in the research people who are doing the PhD, but other things it can also be used for their benefits, right? So descriptive statistics includes the firstly mean, median, mode that we all learn in our class 10. Variance is standard deviation, range, quartile range, and data visualization. Okay? So I'm sharing one software. If it is visible, do let me know the screen of the software. Is it visible, ma'am? Is it visible?
Yes, ma'am,
Yes,
it is
ma'am.
visible.
Thank you, thank you, thank you. So it's a PSPP software. It's a like it's just duplicate, like we can call the SPSS thing. So it's a free. Then we have to pay huge money for having the SPSS software. So this is a free software. So if you all want to download it, you can easily download it. I'll just share how you can download this software, because statistical, this is my topic now. It's a very practical topic, right? So with the helping of understanding the softwares, we will learn how to do these statistical things, and very easily. People might be afraid if they want to write their chapter 4 data analysis, so they are having some other people like paid thing. So we should not go, because it's not ethical. So like that. So this is a PSPP software. It's a GNU project free software, right? So you can easily download from this website. After downloading installation, this it looks like this. So it is having a 2-tab data view and the variable view. So in this data view, we enter the data. And suppose you having a 10 people data, and you want to do the just mean, median, and mode simply. Okay, and if the students are if joined, or the participants who are presenting their paper, they have also having joining through this link. Do let me know if you have any query related to that, because this conference, the motive of this conference is to make understandable, because this kind of workshops, and is separately a paid workshop, but in the we should say thank you to the organizing team. So they invited me as a speaker or a resource person in this conference, so all will be benefited. So feel free to ask anything, anytime. You can stop them, says I'm not getting it, please explain. I'll definitely explain, right? Okay, so this is a data view tab. In this, we entered our data. Suppose one question is that what do you like? You like whether you want to go to visit zoo. Okay, so I'm asking the yes or no from the 10 people. So 1 is for the yes or 2 is for the no. So suppose people 1 he said ki, yes, I want to visit the zoo. So I'm giving the number 1 because we have to codify all the data. 1 for yes, 2 for the no. So second person he said ki, no, I don't want to visit. Second, I'll give the 2 for the no. Similarly, so on. Just randomly, I'm putting the numbers. So this is the data of the 10 people. Okay, this is a variable 001. But what we are 001 say, it says it's a opinion or zoo. We say zoo, yes, no kind thing. So we can write by and no. And when you are entering the variable, the software SPSS or Stata or any software, we have to write the underscore instead of putting the space like that. When I have entered this zoo thing, it is already been published in the data view tab. See, like this. Okay, so this is the data or yeah, alignment right scale here. Input. It's role input output. What role is it? You can automatically do that. Okay, so I want to calculate the descriptive status. Is what I have shown in my slide. The descriptive status says that the mean, median, mode, variances, SD. So just go to the analysis. After the descriptive statistics. In descriptive, you can find number of things, frequencies, descriptives. So descriptives, I'm selecting descriptives. Okay, selecting this variable, putting in the variable tab. What do you want to do? Mean, median, mode, just select it. It has already been selected. Mean, median, mode, range, standard deviation, variance, kurtosis, skewness, anything you want. Then press OK. After putting it OK, another output tab has come. This is output tab. So you can see 10 is a number of people, mean 1.3, standard error of mean 0.15, SD, range, skewness, all the things will be, you can done, because it's a data of 10, you can done the 1000 data, number of responses, you can evaluate very easily, right? So this is a one test we did it. After that, if you want to know the frequencies, how many people are saying no, how many are saying yes, then selecting it again. Then if you want to make some charts, you can also have that facility in this. Draw histogram, draw bar graph, draw pie chart, like that. Continue. And if you want to frequency tables, always okay. Continue. And then press OK. See, now I said 1 for the yes, 2 for the no. It means 7 people are saying yes, 3 people are saying no, means 70% are saying okay, I want to visit zoo. 30% are saying no, I don't want to visit zoo. Okay, this is a histogram. This is a very good presentation of the data, bar graph, all like that. So till now, because we did a very small thing, so any doubt till now, please ask. Feel free to ask anything if you want to ask. Okay, I hope everybody is getting, so I'm moving ahead. Okay, another thing, what the PPT says, okay, following the order, mean, median, mode, we did it, standard deviation, variance, we did it, range, data visualization means chart and the bar graph, we all did it. Okay. Now, another thing is says inferential statistics, estimation, hypothesis testing, confidence interval, decision making, uncertainty. So we'll do that. Okay, so hypothesis means we generally say 2 hypotheses, which is null and the alternate. Okay, null says a researcher always try to reject the null hypothesis and accept the alternate one. Means if suppose I'm saying this is a data of the 100 people and I want to know, because I think this is a healthcare, people are more enjoying in this conference. So suppose you are testing when a person entered in your clinic and 100 persons entered in your clinic, and you tested their BP, sugar, and all the things, right? And after that, after 10 weeks or 1 week treatment, you are again measuring the thing. They are, you told them to follow my diary chart and all. So after 10 days, you are evaluating their results, right? So you want to you want to check the difference between the before the adoption of the proper course and after the adoption of the proper course, right? So what is the difference? So if we say there is no difference, there is no change in their before and after thing, and there is this change. What change? This can be good, this can be bad, right? Or we can say this is a significant change. It can be bad or good, right? So for that, there is no change. We say null hypothesis. Definitely there should be a something change in their life. So that's why we always try to accept the alternate hypothesis, which means there is a significant difference between their before lifestyle and the after lifestyle. And we always try to accept the alternate one and reject the null hypothesis, right? And on what things we judge this kind of results? It's based on the significance level, p-value. And number of my friends, or I have spoken to a number of people, they are after having their PhD, I respect their PhD, but still they are very much confused about what is the significance level and what is the p-value. So in this conference, anybody wants to volunteer what is the p-value and the alpha value? Anyone can tell me what is the p and what is the alpha value? Because we have to do the one. I'm a resource person, but if you do the interaction kind of thing, so it will be very much easy to make yourself understandable. If anybody wants to volunteer or anybody want to say, ma'am, this is a p-value, this is the alpha value. Anybody? It's an open platform. It's a conference, so we can have words with you all. Okay, no issue. So p-value and the alpha value means, just a minute, I'll just write it down here so that everybody can understandable. See, alpha value is a table value or a standard value. By this, we can compare the calculated value, which is called the p-value. So this is a basic difference, right? I'll just tell you with the help of example what alpha value says and what the p-value says. And significance level, the level of significance, the percentage, generally people check with the 5%, with the 1%, with the 1%, and with the 10%, right? So most of the PhDs are done on the basis of this 5%, right? So if we are saying we have to check our results on the 5% level of significance, it means our alpha value will be 0.05. So our p-value, which is being calculated, so this value should always be lower than this. If your, make me another slide. If your p-value is 0.02 and your alpha value, which is a standard value, which is your 0.05, so this value is lower than this 0.05, it means you can reject your null hypothesis and accept the alternate one. And if your p-value is, I'll just tell you with the help of the software, so you all be understandable with this. Yeah, 0.088. So this is greater with this 0.05. It means you have to accept your null. Means there is no significant difference. Yes, so you have to reject your alternate and accept the null. So I'm just also writing if you want to take the notes. So in this case, in the first case, your null hypothesis will be rejected, and your null hypothesis will be accepted in this case. Then, So I'm doing one test on this data. Okay. So this is the data I've just taken out. Okay. So now I want to ask that whether you like research methodology. You know, I'm asking with the 10 people, if you are if you like very much, so you get the 5 point. If you like okay, okay, I like that, so you will give. I'm okay with it 4. You are not pretty much sure good or bad, so you are giving. You are neutral. Your opinion is neutral, so you are giving number 3. And you say no, I don't like, so you will give number 2. And if you say I seriously, I'm totally disagree, I seriously don't like, and I'm very much not comfortable with the RM thing, so I'm giving 1. So 5, 4, 3, 2, 1 is the Likert scale. So number of people make the question on the basis of this Likert scale also, which is called the interval scale actually. So 5, 4, 3, 2, 1 is the range. So respondent 1 says, I like very much. Respondent 2 says, I don't like. Respondent 3 says, I'm okay with this. And respondent 4 says 4. So accordingly, we are giving the coding simultaneously. 2, 5, 5, 4, 5, 5. So this is the result. So this is the data. So now I want to test whether this is a significant or what is this. If I want to check, so I can do one test which is called the one sample t-test. It's very much used. So I'm just clicking on one sample t-test, and I'm selecting this. Oh, sorry, I haven't given the name to variable 001. So this is a preference or about the research methodology. So I'm giving the RM. Reference. Okay. So I'm testing it like this. Compare means one sample t-test. Okay. Okay. Analysis. Compare means one sample t-test. And then, okay. So in the one sample t-test, we always use, because number of the research have been found, if you are choosing the one sample t-test, you have to give some standard test value. So generally, researchers use 3. This is the mid value of this. So then press OK. Then analysis, compare means, okay, I already did it. So this is the result of one sample t-test like this. So you can see this is the 10, which is total number of the sample size. 3.9 is the mean, and your significant, this is a p-value. So what is this value? It is a 0.081. This is a 0.081, and I'm checking with the 5% of level significance. How come? Because it is written 0.95, right? So as we will go to the PPT, if your alpha value should be lower than the 0.05, then only you can reject your null hypothesis, right? But in this R case, it is 0.081. It means we can't reject our null hypothesis. We have to accept our alternate one. So by this, you can judge your statements, right? Because why it is coming? Because we have only the 10 number of sample sizes 10. So this may be the reason. So accordingly, we do the test. Till now, if you guys have any problem, any suggestions, please let me know. Otherwise, I'll continue. Anything you want to ask, please tell me. Okay. Okay, so I'm thinking there's no confusion or no queries. So
excuse me, madam.
yes.
This we can do only this software only, na? This
Ma'am, we can do this through the PSPP or through the SPSS, through this Stata. So like that we can. And suppose if somebody doesn't have this software, so we can also perform this thing in the Excel also. Okay, so good observation, ma'am. So I'm also sharing this Excel thing. So this Excel file is visible?
Yeah, madam, it's visible.
Okay, fine. Thank you. So in this Excel, we have the facility with the grace of the Microsoft. So we go to the file option, then go to the options. Then there is a one add-in is there, add-ins. Okay. In this add-ins, we found analysis tool pack, analysis VBA. We just select this analysis VBA, and after selecting it, then press OK. When you press OK in this data tab, you will find the data analysis tables here, right? So same thing what we did in this software, we can also perform all the tests in the data analysis. Yeah? So suppose I'm asking the same question with the people, like what you think from this RM course. So suppose one correspondent is saying 5, another is also saying 5, 4, 3, 2, 1, 4, 5, 4, and 5. So this is the data, right? This is the data of 10 people. Now the same thing what I did in the software, I'll do in the data analysis tab. So I want to firstly do the descriptive statistics. Selecting the descriptive statistics, then press OK. What is your range? So this is my range. Selecting this upside arrow, and this is my range. After selecting the range and where you want to get your output, so I need my output here also, here in the blank portion. So selecting this blank portion, I need my output here. Select it. And then what you want to know? Summary statistics I want to know. I want to know the confidence level, kth largest, kth smallest, and then press OK. After doing this, all the things, so this is the result. So see, 3.8 is your mean, SD is your standard is 0.44, and with this data you got to know that the range is 4, minimum value is 1, maximum is 5, sum is 38, count is 10. So like this you can have an idea about your data, right? So this is clear to everybody? Okay.
Yes, madam. It's clear.
Okay.
Thank you, madam.
Thank you so much, ma'am. Thank you for giving this idea. Actually, I totally forget to explain with the help of the Excel. Thank you so much. Okay, so one sample t-test, we did it in this software, and another thing we will do the same thing. Yeah, if you want to prepare the histogram, so histogram, then press OK. Then input range, this is the input range. Okay, and okay, there's no bin. Okay, so we will create another data. Okay, so we can leave this, and very importantly, another test, if you want to test the two things, right? Before and after, like I have just put it an example, before coming to the clinic, what was your results? After going to your home, you know, what is your results? So if you want to compare the two things, suppose after before going to the clinic, your the 10 people's weight was 50. One people weight was 50, second people weight was 45, and 55, 60, 65, 67, 63, 76, and 81, 91. This was the weight of the people before going to the clinic, and after having the clinical experiments or following of the diet chart and all, the weight of the first people 50, whose weight was 50, now his weight is 49. 45 people, 45 weight of that people, his weight is now 48. 55 is 50, 60 is 55, 65 is 63, 67 he has gained the weight, so 70. 63 is 65, 76 is 70, okay, 81 is 65, 91 is 79. So this is the data, means that is there any difference so I want to test ki after adoption of that treatment in the clinic, is there a difference between the weight, right? Because some people gain their weight, some people less, their weight has been decreased. So I want to know that is there a difference, and what difference is the significant? So always use the term significant in the research kind of thing. We always use the word significant, right? So if there is a significant change in the weights of these of this treatment, is there a significant or not? So we'll check with the help of the pair t-test. So data analysis, we go to the pair t-test, select it, okay. And in variable 1 is this. Variable 2 is this. Labels, alpha value point output. Okay, new worksheet will get the result in the new worksheet. Okay. So this is the result. So you can see the p-value is 0.046 and another p-value is 0.09. So in this first case, which is on the one tail test, this is significant. It means yes, after having that treatment, there is a positive kind of thing. It means there is a significant change in their weights on the basis of the one tail t-test. Two tail says no, there's no. So if there is something we are getting, so we can take this kind of data also, only this for your paper and all, right? So whatever we did today, right? So if you are putting this kind of result and make some charts and bar graph and another one more frequency test you do, so you can prepare your paper within 15 minutes. Your data analysis part has been done in 15 minutes, and you can do your intro LR within a week. So within 15, it's a maximum time I'm saying. Within 15 days, you can prepare your paper, research paper. And it's a very good paper because you are doing your test and all by yourself, right? Not by the paid person. So you can conclude that yes, there is a difference between the after having the treatment from that clinic, following that diet chart. Yes, there is a significant difference. So this is the data of the 10 people. You can have the data of the 100 people also. So you can write your paper, you can write your own thesis, you can do all your analysis by yourself. It makes you're comfortable because after having your PhD, you can be the guide of someone, right? So do it by yourself, not outsource. Okay, so this is done, and the same thing I'll also do in the PSPP, this pair t-testing. So if your data is in the Excel, you can just copy the data from the Excel and you can just put in the PSPP also. It's so easy, just copy and paste. So variable 001, it's the data of the before test, and this is after test. Okay, so running the same thing, and this compare means pair t-test. So this is a before and this is after. Okay, this is after options, 95% perfect, and okay, so what once it's not coming. Okay, let me check. Okay, just a minute. We'll check the scale is correct, nominal. Okay, it's a nominal. Then we'll check paired sample t-test. Okay. After, okay, we'll check one by one. Oh, sure. So, okay, pressing this like this. Okay, just a minute. Now it will come. Analysis, compare means, pair sample t-test. Okay, removing this. Okay. So, this is like this. Yeah. So before and after, see, it is giving already. I don't know what's the problem with that. It's giving. So before and after, it's saying correlation is 0.91. 9.911. It's a significant 0.00, right? And this is a mean. The mean value of all this before and after. This is standard error. Okay. So SE is this, and in our Excel, what was the results? Okay. So it was where it is, it is here. Okay. Okay, I think I have closed that. Okay, but no issue. Like this in the PSPP also, you can perform the same test what we performed in the Excel. So this is a paired sample t-test while with the comparison of the before and after having that therapy or that diet chart, and you can do the mean, median, mode, frequency, SD, skewness, kurtosis, all the things. Okay, so what we have learned so far, we did this. Okay, now the z-test and the one sample t-test also we did it. Okay, when your data size is more than 30, right? When your data size is more than 30, you go for the z-test, right? And also when you are aware about your population standard deviation or population variance, then also you can perform z-test. But generally we see if your sample size is more than 30, then you go for the z-test. And pair t-test we did it. ANOVA is very much used in the healthcare thing. So in this we have to check the between the group and within the group. Okay, so just quickly we'll do because we are running short of time. So I'm just performing the one ANOVA test. Okay, so analysis, one way ANOVA. So RM pre I'm just taking. Okay, so we need to do this like this before test. And factor after test. Okay, and just I'm performing this descriptors, homogeneity, then okay. So this is their ANOVA results. So because this is a between group and this is a within the group, you can see this a significance value is 0.340. It means this is not significant because we have a less data. But like this, you can perform this ANOVA test, one-way ANOVA test in your paper, in your thesis. And if you want to perform reliability, it's a very important thing. Reliability means when you prepare your questionnaire and you want to know the questionnaire, the questions and the responses are reliable, means we have to check the validity and the reliability. Reliability means the consistency between the answers, right? Because whenever you are giving the questionnaire to someone, he just write the five, he's just giving the 5 5 5s throughout the questionnaire. This is wrong, right? So whenever you want to check the consistency, this is the first step in the research. So before applying all the tests and all the things, you just check the reliability of your data. So suppose this is in this for this reliability checking, we have to check the questionnaires reliability. So I'm just randomly giving a number 5 4 3 2 1 1 4 3 2 1. Okay. So this is for the reliability. So reliability 2, and this is for the reliability 1. So I want to check this responses are there's a consistency in the answers or not. Just go to reliability part and checking these 2 items, because these 2 items are related to particular factor, because one factor consists number of the variables. So if we want to check the reliability of a factor which contributed in a particular factor, so we have to select all the variables which comprises one factor, right? So then okay, now we check the reliability. So this is reliability. This is quite low, means 0.27. There are 2 items. This Cronbach alpha value at least more than 0.5, right? So this is 0.27, and suppose I'm writing another variable. Oh, sorry, I haven't write one. Okay. So if you want to check the reliability, suppose this contains the three variables in a factor. So this is a response, and I want to check this reliability of this factor, which comprises 3 variables. Okay, so analysis, then reliability, then selecting this reliability 3, and we always take this show descriptives for the scale if item deleted. Means suppose in a factor there are 3 variables. If I'll delete what item should be deleted so that our cron batch alpha value will be increased. So just check this checkbox, and we going to this tab. See, our cron batch alpha value is 0.6, which is quite good, and it says ki if you delete reliability 1 variable in your factor, so your cron batch alpha value will be 0.93. So we can do also, we are not taking REL1 in for our paper on for the questionnaire. So I am not taking REL1. Just check 0.93, whether it will come or not. So going ahead, suppose I am removing this variable. Now I'm checking. So here it is, 0.93, right? This is good, right? And it's saying no, there's no need of deletion of any item. It's quite good. So this is the value of the cron batch alpha. It means our REL2 and REL3, they are very, very much good variables for your particular factor, right? So only 1 minute is left. So I'm just briefing all the things what I have explained so far. And if you guys, if you have any doubt, please ask me the question. So what we did, we learned mean, median mode. We did bar chart, pie chart, and we also did the one-way ANOVA sample test, pair t-test, and we did ANOVA. We did all the things in the Excel also, and lastly we done the reliability, right? So don't be depend. So concludingly, I'm saying don't be dependent on someone else. Do your things because it's your research, it's your thing. So do it by your own, not by someone else. So this is all about today. Thank you so much, and thank you so much for giving the opportunity as a keynote person in this esteemed conference, and especially Hemant sir. Thank you so much. Thank you, Manisha ma'am. Thank you so much.
Thank you, Kritika ma'am, for this lovely session. Because even we have attended many conferences and we have tried to learn this process, but still, whenever we do the calculation and all, definitely we get some error. Not some, like most of the time, we used to get error, ma'am.
Right, I know.
Seriously, how smoothly you have explained the things and all to us. Thanks a lot, ma'am.
Yeah,
for
so
this.
thank you so much, ma'am. Manisha ma'am, I'll request to the organizing institution, so we can do number of workshops like that, so everybody can be benefited with this. And the next, we will meet in the next seminar.
Definitely, ma'am. And so
Thank you
we
so much,
will
ma'am. Thank you.
most welcome now. So
Thank
our
you.
next speaker is
Hello.
yes.
Hello, Madam, can I ask one question?
Please,
So,
please.
Madam, for the reliability, we have, for example, we have in the preparing the tool, there is 30 questions. So, can we divide it into odd number or even number? How we can take this?
So 30 questions. So what you do, your questionnaire have 30 questions, right? So these 30 questions means like there is some part A, part B, part C, or whether it is in the like some 5 questions related to one particular theme, another 5 questions related to another theme. So like that.
So like that, we have to, it means you are doing the reliability 1, reliability 2, reliability 3, like that we have to do.
Right. What do you do,
Yeah,
ma'am?
okay.
What do you do? You just check the theme. So suppose your 5 questions related to particular theme. So divide the questionnaire according to the theme, like part A, part B, part C, and all the questions which comprises in the part B. So giving the number like REL, RE1, 2, and check the part A reliability togetherly, part B separately, and part C questions separately. So then you will get the results.
Okay, madam. Thank you. Thank you, sir.
Okay. Thank you, ma'am. Thank you. Thank you.
So we will move forward with the second guest speaker, and she is Dr. Chinna Devi, ma'am. So let me introduce Dr. Chinna Devi, ma'am. She is a principal of MG University, Kishanganj, Bihar. As her qualification, she has completed her PhD in nursing, and her as her postgraduate and PhD research experience, she has served in many colleges, and she has willing to say that she's having the specialization in child health nursing, and she has guided many of the student in as for the PhD level and for the UG and PG level. And let me tell one thing about Chinna ma'am, that in this age also she is very enthusiastic, and she is very keen to learn new things over here. And even I have also learned many things from Chinna ma'am. Only once I have met her, but definitely I would like to meet Chinna Devi ma'am again and again for learning many more things. It's over to you, Chinna ma'am.
Thank you so much, Manisha.
You're most welcome,
Good
ma'am.
morning, everyone. This is Dr. Chinna Devi. I think no need to introduce myself here. Already Miss Manisha have introduced me. I'll straight away go to my topic.
Ma'am, you want me to share your topic?
Yes, please.
Okay, sure, ma'am, I will do it. Let's see.
Manisha, I have shared.
Okay, ma'am.
Okay. Okay. So my topic is AI powered research and personalization.
I think ma'am, she's facing some network issues and all. Yeah, let's see. I think ma'am, she's facing some technical issue over there. So we can move on to the next.
Manisha? Hello?
I think you're having technical issues from your end, ma'am.
Am I audible now?
Yes, ma'am, you're audible.
कहाँ से मेरी आवाज नहीं सुनाई दिया?
Ma'am, from starting only, ma'am.
From starting. Okay.
Yes, ma'am.
My slides?
Ma'am, it's not visible.
Slides also are not visible. Just a minute, ma.
Yes, ma'am.
Okay. My network is low. I will come from mobile, ma. This is my laptop. Okay. I opened. Now I will open my mobile.
Yes, ma'am. Okay, okay, ma'am.
Okay. Manisha.
Yes, I will share your slide, ma'am.
Okay.
Yes, ma'am. You can continue, ma'am. I already have shared your slide.
Okay. Okay. Introduction. Resound array. Hello.
Yes, ma'am.
We are not merely witnessing a technological revolution. We are experiencing a redefinition of intelligence itself, where machines do not just compute, but learn, adapt, predict, and personalize. So the era of intelligent research, traditionally previous days the research was time consuming, labor intensive, data limited, and today AI enables us automated literature reviews, predictive modeling, pattern recognition across massive data sets, real-time data analysis. AI is transforming evidence synthesis, clinical trials, optimization, population health analytics, nursing research. So AI doesn't replace researchers, but it augments human intelligence. What is AI powered personalization? It customizes content and services for users based on behavior, preferences, and history. We can utilize AI powered personalizations, improves user experience, and we can use in predictive algorithms. From data to personalized intelligence, personalization is the new gold standard. AI now enables us in precision medicine, individualized patient care plans, adaptive learning platforms, predictive risk scoring. So here in this model, we can see that how we can utilize AI identifying high-risk patients, electronic health records, improve patients' monitoring, preventing medication errors, reduce fraud, waste, and abuse, reduce costs, reduce readmission rates of patients, improve patients' outcomes. So in healthcare, AI is utilized. AI, artificial intelligence, analyzes genetic, behavioral, and clinical data. It predicts database progression. It supports clinical decision making. In education, how we are utilizing artificial intelligence is it adapts learning pace, it tracks performance patterns, it identifies gaps instantly. So the era of one size fits all is over. Now the era of precision personalization has begun. AI in healthcare and nursing research. In nursing professionals and healthcare leaders, we must recognize how AI supports in research. So AI is supporting in early detection of high-risk patients, remote patient monitoring, smart documentation, burnout prediction models in nurses, clinical decision support systems, so that we can identify the problems and we can go for research. This is not futuristic, but this is happening right now. AI and research methodology transformation. AI is shaping research methodology in automated data cleaning, real-time statistical modeling, AI-driven SPSS, and research integration, predictive analytics, nurse-led practice-based qualitative data analysis. So systematic reviews that once took 12 months can now be assisted in weeks. So that much time we used to use before for the research. But nowadays, within weeks, we can complete our research through the help of AI. Meta-analysis, bibliometrics, and citation mapping are becoming smarter and faster. So ethical responsibility in artificial intelligence. With power comes responsibility. We must address data privacy, algorithm bias, ethical artificial intelligence governance, transparency in research, human oversight. AI must remain in assertive but not authoritative. We have to understand this way. It is not assertive but authoritative, ethical but not exploitative, inclusive but not biased. Technology must serve humanity but not control it. Technology should not control us. Future directions are in the next decade, we will see AI generated hypothesis testing, autonomous research systems, real-time global research collaboration, personalized health ecosystems, AI integrated nursing practice standards. Countries that integrate AI into research policy will lead global innovations. Those who are not going for artificial intelligence help, they will fall behind. So a call to action. As global researchers and healthcare leaders, we must integrate artificial intelligence literacy into curriculum. So we have to make artificial intelligence, we have to integrate into our curriculum, nursing curriculum, encourage interdisciplinary collaborations, develop ethical artificial intelligence frameworks, promote evidence-based artificial applications, invest in AI-driven research infrastructure. So AI is not a threat. It is a tool. It is a powerful tool. The question is not, will artificial intelligence change research? But the question is, how will we lead that change? Next slide, please. So as a summary, let us not fear artificial intelligence. Let us shape it, guide it. Let us ensure that artificial powered research and personalization create a world that is smarter, healthier, more equitable, more compassionate. Because in the end, the true intelligence behind artificial intelligence is human wisdom. So at last, we ourselves have invented artificial intelligence. So how far we can utilize that, we have to think. Thank you. So that is all about my presentation. If you have any queries, please.
Good morning, madam.
Good morning.
Madam, can we use the AI for the research, and how it is the reliable?
Definitely we can use AI in our research. Like in foreign countries also, they are encouraging nowadays to utilize in our research. There are many websites where you can go and utilize for your research. But completely you should not depend on AI.
Yeah, madam, because the we are doing in the software. So,
Yes.
for example, the Excel or other software also. And if we are in the placing into the chart GPT or any other tool like the AI tool, so we found the many of the difference or error occur. It means the difference of the test or statistical analysis is found there. That's why I'm asking.
Yeah, but exactly you have to upload your data. Definitely you will get accurate results, but sometimes, completely, that is what I'm telling, that we should not completely depend on AI. We also
Yeah,
have
madam.
to,
Yeah.
yes,
Yeah, madam. Yeah. Thank you so much.
thank you.
Thank you, Chinna ma'am, for this lovely presentation. So, definitely, in the research area nowadays, that AI is taking places AI is taking the human places. But what we should do exactly, that we are not supposed to fully dependent on AI, because sometimes,
Exactly.
not sometimes, most of the times when we use the what you can say, when we use the websites and all, we used to get lots and lots of error, because nowadays most of the research papers, they are totally dependent upon the AI only. We have seen even whenever we review the papers also, ma'am. So does it makes that the people, those who are using the this AI, are they totally lost out, or like what, their brain has gone shrink totally, because they are not able to use their intelligence totally, they are dependent upon the machines and all.
True.
So we will move to the next speaker. Our next speaker is Ankita Jain ma'am. So welcome, Ankita Jain ma'am. Sorry, Ankita Garg ma'am. Okay. So
Yeah, good morning. I'm just connecting me.
yes, ma'am.
Just wait for me 2-3 minutes. Actually, my time was scheduled at 11:30, so I was in lecture. Just now I have finished it. So I'm just
Okay,
opening and
ma'am.
sharing
No
my.
problem for that, ma'am.
Yeah. Please give me an access to share the screen.
Yeah. So before that, I would like to introduce Ankita Garg ma'am. She is a professor in Department of Chemistry, Sanjeevani University. And as a so she has written many. She is having research interest in heterocyclic synthesis and multi-step reactions and multi-component reactions, green chemistry, and drug design and molecular modeling. So definitely, we are going to have a very great session with Dr. Ankita Garg ma'am, because she's from the chemistry department. So even she's having the dual specialization in MSc. Oh, one is physical chemistry and second is the organic chemistry. So definitely we are here to learn many more things from Ankita Garg ma'am. It's over to you, ma'am. Ankita ma'am, if you want, I will share your PPT, ma'am. I'll do.
Hello, Manisha. Good morning.
Good morning, ma'am.
Good morning. So, by the time, can I just take a lead?
Definitely, ma'am, we can have some conversation also over here. Like we are having many more experts.
Okay. Okay.
Ma'am.
Okay. Thank you, Manisha.
Yeah, most welcome, ma'am.
So, I'm Dr. Sarika Saxena, organizing. Can you hear me?
Yes, ma'am. We
Okay.
can hear you.
Okay. Thank you. I'm organizing chairperson of this international research related this conference on healthcare and educational research. And as I've seen the yesterday program, all the presenters, they have presented their poster and they are the research under the supervision and guidance of the team of the GIAR. And the judge has taken a very, they have made a very good conversation with them, asking a question. And I think the most of the participants, they are also get the knowledge and they are very well aware of their topic also. They have communicated nicely. And today, the session of this for the speakers, and we got a very good speakers. As I have seen the list of Dr. Sakshi Chaturvedi, Dr. Kritika Tekwani, Dr. Ankita Garg, Dr. Samanthan Thul, Ms. Ria Jalal, and Dr. Fizrana Begum, and Dr. Ruchika Rani. And I am really very grateful to you all, everyone, those who are taking many these efforts for taking a participation into that as a means to share your expert talk. So I would like to congratulate each and everyone and listener those who
Am I
are
audible to you?
the
Yes, ma'am.
Uh, you have
But your
not given
voice,
me the access to share the screen. I want to share my screen, but you did not give me access to share the screen.
ma'am, just try it again, ma'am.
Are you sharing my screen, my slide?
Yes, ma'am, I will do it, ma'am.
so Manisha here I am binding because I think she's ready.
Yes,
Uh,
ma'am.
so anyway, I would like to wish you all the speakers of the today's and the tomorrow's event speakers and many congratulations to each and everyone. Thank you so much for giving your time and connected with us with the GIAR. Thank you so much. Thank you.
Thank you. Over to you, Ankita, ma'am.
Yeah. Are you sharing?
Yes, ma'am, I already shared, ma'am.
Okay, okay. So good morning, everyone. Am I audible?
Yes, ma'am.
Am I audible now?
Yes, ma'am.
Okay. So here we thank you very much for the organizers because you have been invited me for this prestigious conference, and here I'm presenting something about my research. And actually we have to interconnect now two or three fields, then only we can get something substantial in our hand. That's I wanted to just address in today's lecture. So you see, here I have taken a topic of bridging organic synthesis and biomedical research, an integrated approach to drug development. What happens generally that next slide, would you please give me? What happens generally that whenever we are thinking about the synthesis of organic compounds, then sometimes we have a gap that we have synthesized. Suppose very good scaffold we have synthesized. Then what are the application part of that particular scaffold? That you have wasted so many chemicals, you have wasted so many resources, and other than that, we have been harming the environment also. If we are doing any chemical reaction or something, then what are the application part of that particular research which we are doing in lab? So there must be some application part, and the best application part which organic synthesis has is the drug discovery. If we are synthesizing some drug, that means we are doing some good job. But sometimes it happens then when we are going for in vitro means when we go for lab testing of the compound, when we go for in vivo means we are going to get further clinical trials and all, then there might be some problem. So today, because it is a long process, it is very costly process sometimes, and again it is harming the environment as well as to other things. So today we are going to discuss that how we can integrate the computational methods with the organic chemistry as well as with the in silico methods can be clubbed with the in vitro methods and in vivo methods. So a little bit background I want to give you that how we have been reached till here. That earlier how we were synthesizing the drugs, what we were doing previously in research in organic synthesis also, and in the drug discovery where we were previously and now where we are standing, and what are the future prospects of our drug designing. So I want to share some insights about it. Would you please share next? Next
Yes,
slide,
ma'am.
ma'am.
Yes.
Ma'am, please share the next slide. Should either give me the access so that I can share my own. Is it visible now to you? My screen? Is it visible? So I myself has opened my slides. So is it visible to everyone? Would you please answer? Okay, so here we have evolution of drug discovery or the background of the drug discovery. Earlier in the ancient time, what we were doing, we were looking for these kind of medicines which we can, if we talk about in Indian context, it was Ayurveda. Still we have Ayurveda, but now most of the people are going for allopathy, and we have been taken a long path actually. Traditional Chinese, Egyptian, these are just for an example. Then further we were developing a few allopathic or modern medicines. Then after what happened, the trial and error era was coming. It means within 1950 to 1990. That means what? That we were at large scale, the companies were trying to synthesize so many drugs, but there were so many diseases which were happening not only pan India, but worldwide. So many diseases we were listening at the time of 1950s, 60s, 70s. But now those diseases are just gone, just because of vaccines, because we take a vaccine and that disease is not even existing today. And then after 1990 to 2005, we were having target based drug discovery, in which we were targeting a particular disease. Because although we are doing so much of development, but with the development, what is happening that with the development, so many other things, means we are harming the environment, we are changing our lifestyle. So disease are also just coming in very different way. So this is also a problem, that the disease are also in a way that we are facing so many other things, which we have to make a target, that which area and which part of the body should be targeted, which particular point should be targeted. High throughput and combinatorial chemistry also, nowadays these two things are together, organic chemistry with the drug designing. So organic synthesis, whosoever is organic synthetic chemist, they are synthesizing something by asking with the biology people, by asking with the bioinformatics people, by asking with the scientists who are working in the drug designing, that which kind of compounds we should design, so that they will be having a very good application in the drug designing process. So now we are in present to future. What is that integration of the computational method? This is actually the today topics discussed, that we are integrating the computational methods with the other methods. So what is it? What are the computational methods? In the computational methods, we have many kind of methods who are working only in the software through AI and through other softwares. We are using those methods. We are just checking the efficacy of those compounds, that what we are going to synthesize, whether they can be, there is a prediction of drug designing, that whether they can become a good drug or they cannot become a good drug. So if they are giving positive result, then only we should proceed further, otherwise we should not. But this approach is also having a few problems, a few shortcomings, limitations. Those limitations also we are going to discuss. But we will see that results also, when we just go for the computational methods first and then go for organic synthesis, and after synthesizing the compounds, we will check for in vitro and in vivo and clinical trials, then it is saving very much of time, energy, resources. We will check the data also. I have been summarized many data in this particular slides. So here, if we took this particular slide is about that, how do we go further? So first of all, sometimes previously people were doing molecular designing, and then after they were doing organic synthesis, then after in silico, in vitro, this. Previously people were doing this kind of things, but now they are doing what? First they do in silico screening, then only they go for organic synthesis, and then after in vitro and other clinical trials, so that many resources, many time, and too many things can be saved. So you see here we have a compound in which there are structural activity relationship. What does it mean? That there are many tools, in silico tools, through which we can design our compound, that this particular compound must have nitrogen over here, oxygen over here, or any other sulfur heteroatom should be here, so that the lipophilicity is going to be increased, or binding is going to be increased, or toxicity is going to be reduced. These kind of parameters are there, so many, largely there in the research papers, in the literature. So we have, whosoever is working in that direction, they just go deep and deep inside the research, and then only they design their target, then only they design their organic compound, so that many resources can be saved. Now you see that drug discovery has evolved from trial and error approaches to rational and technology driven research. Earlier, we were doing trial and error approach. We were just going in the lab, we were hitting the chemical reactions daily, 100, 200, many students, they were just going for 50 reactions daily, and they were getting only one or maybe sometimes none of the reaction which was successful. But now we are taking rational and technology driven approach, so that the resources can be saved. Modern pharmaceutical research requires collaboration between different fields, so that a concrete thing can be driven. Data integration, the data integration is very important, because everything is, all the softwares we are using, those are also AI compatible. So data integration is also very important, and multidisciplinary expertise. Even I am an organic synthetic chemist, still I am using all the in silico methods by my own. I'm not using anybody, I'm not taking anybody's help to do those in silico methods. Why? Because I cannot wait for this much time. If I want to synthesize something, I do not have unlimited time to wait for somebody. So whosoever is there in our lab, students, those are also very much compatible to use these in silico methods by their own. Even if they are organic chemistry students, they are doing bioinformatics field by their own. So that's known as multidisciplinary expertise, which we need. Then only we can be benefited. If we talk about global drug discovery landscape, over 30,000 drug candidates is screened annually. You see the data, and only 1 or 2 reach in the market. These many drug candidates, every year people are screening. They are going for in vitro, sometimes they are going for in vivo, sometimes they are going for even clinical trials. Still there is a very long process, 15 years, 20 years sometimes, and only 1 or 2 they reach to the market or for the commercialization. High attrition in the clinical phases also. There is very much, we will see that there are so many failures when they reach at the clinical level. The major causes behind it is toxicity, poor pharmacokinetics, low target specificity, all these things, and integration minimizes these risks. If we integrate different branches of this particular, our conference is about the integrating different fields, integrating different multidisciplinary, different discipline together, so that resources will be saved and we can get the result. Even if we are wasting resources, suppose, but we are getting something, then also it is okay. But we are wasting also and we are not getting anything, that is really very harmful. So if we are getting something that is needed now, that different disciplines should be integrated together, so that the compound which we are synthesizing, which we are testing, that must not be toxic. Suppose we are taking a medicine, suppose we are taking medicine for cough and cold, and that medicine is so toxic that we have been developed some other kind of disease in our body, then who is going to give the permission to that particular drug? So these are the problems when somebody is designing drug, then these are the issues which we have to address. Now if we talk about role of organic synthesis, what is the role of particular organic synthesis? Why we are talking about too much organic synthesis? Because literature has shown that heterocyclic chemistry, the heterocycles, I did my PhD, my postdoc and everything in this field only, heterocyclic chemistry, medicinal chemistry field. So heterocycles, they dominate the modern therapeutics. Whenever you open any drug, there will be heterocyclic atom inside the ring. Structural diversity that improves the bioactivity. If we have a diverse structures, then bioactivity will be improved. Functionalization enhances selectivity. Whenever we see multifunctional compounds, if the functional groups are increased, the selectivity will be increased. So it is complete literature which is available, and it is the matter of study. Whenever a researcher comes in the field, the PhD scholar or anybody who comes in the field, they first check the structural activity relationship. They check that which kind of compounds will give you the application part of drug synthesis, then only they go further. If you check about the market, then 60% of the marketed drugs contain at least one heterocyclic moiety. It means heterocyclic atom means nitrogen, oxygen, sulfur, other than carbon and hydrogen. Those heteroatoms, if they are available in the moiety, then we call them as heterocyclic compounds. So if we check them, there are so many. And popular heterocyclic scaffolds, if you say anti-cancer drug, anti-malarial drug, anti-inflammatory, antifungal, anti-parasitic, any drug, if you open, if you check for any drug which is available in the market, if you open the structure of that drug, they will have any heterocyclic moiety, that is for sure. So that's why this field is extremely important, because without organic synthetic chemists, the drug designing people cannot do anything. And if drug designing team is not working properly, then an organic synthetic chemist will synthesize something and it will not result in any fruitful outcome for anybody. So now if we talk about synthetic methodologies, so there are many methodologies which we have in organic synthesis. I have been jot down here, just mentioned a few, like multi-step synthesis. Earlier people were very much concentrating on multi-step synthesis, but now all the companies, they want very short synthesis cycle. So they are researching very hard, they are doing very much of R&D. That's why the path of the synthesis can be minimized as much as possible. So green chemistry principles now they want to follow. So all these things for getting optimized result, microwave synthesis also people are doing now one pot reaction. One pot reaction means within one vessel, from starting to last getting product, everything is done within one pot. So they are using so many techniques earlier which we were thinking only imagination. Those all imaginations become true now in the synthetic methodology. So many methodologies we have, and after synthesizing something, first we have to characterize it. As we all know that through characterization only we can justify that yes, the compound is the same which we have been synthesized. So there are multispectral techniques, NMR, HRMS, many techniques are there. Now we see computational drug design. So in computational drug design, the review report suggests that you see 60 to 70% of the reduction in screening cost. Even if people are just saying blah blah that if you are doing in silico, who is going to believe? It is just a prediction. You are not going to get any concrete result or whatever. Just listen them and see the data. That 60 to 70% reduction in screening cost have been mentioned. This much we have saved. When we are using the in silico method. So why to deny those methods? And it is not taking much time also, because the softwares are very much available now and they are working very fastly. If you have a good computer system, if you have a good cloud computing labs in your even if you are not having cloud space, even if you have only a good laptop in your hand, if you have a good desktop, then also it is very time saving and you can finish the in silico screening very soon. And that faster lead identification, these methods are very fast and they improve the target selectivity also. Many tools like Autodoc, Schrodinger, Swiss ADME, PyMol, MD simulation, many tools are there which are available. If we talk about molecular docking advances, molecular docking is also getting advanced and advanced day by day. Nowadays we are doing many type of docking. What is molecular docking? When we dock our ligand means the compound which we have synthesized through organic method. We have synthesized some drug and now we are targeting the protein that suppose any bacterial protein, any viral protein, any cancer cell or whatever, that how this particular compound which we have synthesized is going to target that particular bacteria, virus or whosoever disease we have, the target identification. So we dock them together. We just interact them together. If they interact well, that means this particular drug which we have synthesized, this particular drug is going to just to save us by this disease, because it is going to kill that virus, it is going to kill that bacteria. So that's how we proceed further via docking. That's called docking. So flexible, ensemble, AI assisted, water mediated, because nowadays people do not want to use even solvent. Green chemistry principles are so prevalent nowadays in the whole world that people want to do reaction merely in water. They do not want to use even ethanol. So people are working in this direction. They are improving the prediction accuracy. More and more accurate softwares are coming in the market, although this time they might be a little bit expensive, but gradually people are getting more and more. ADMET and drug likeness review will give you that whether this particular compound which you are going to synthesize, that can be proven as a good drug or not. They apply many parameters like Lipinski, blood-brain barrier, CYP, hepatotoxicity, immunotoxicity, neurotoxicity, many toxic parameters they just study, means in silico parameters they first predict, then only they give you data. And they are working on numerous compounds which are available in their data already. All the data are preloaded in that software. So they are working on billions, trillions of the compounds which are already loaded in this data, and those data, after just checking those data, they give you the result. And this process is very fast in the software. So that's how we can save our time, resources. And you see, early ADMET screening has reduces the failure by 40%. Failure at clinical trials they are talking about 60 to 70% reduction here in the screening cost, and 40% reduction here in the failure at clinical trials can be possible if we are using in silico methods. And after doing in silico and organic synthesis, then only you should go for in vitro screening. Earlier, what people were doing, they were blindly synthesizing something. They were just going for the lab. There are many labs available, just BLAL or just for the name a few. There are many labs. Those who are working, they will give you a result, this, and just students get published the data, and then just they forget. But this should not be the approach that we should forget. everything. We should go first for in silico screening, then only you should synthesize something, and then go for in vitro screening, in vivo screening, and clinical trials. So you see, in vitro screening, current practices like suppose antimicrobial, anti-cancer, anti-inflammatory, antioxidant, enzyme inhibition, many diseases are there. For all the disease, the lab specific people are there. They are doing their test. They are using many methods I have been mentioned over here. So they are using. So what integrated drug discovery model says? So literature proposes that earlier people were designing and then predicting. Now this loop has been changed a little bit. Now first they will predict, and then after before prediction they will do earlier people were synthesizing and they were predicting. Now they were designing something, some compounds on paper. They are not synthesizing it. They are just designing it on paper, and then after on paper doing it, they will predict it through software, because just on ChemDraw or any other Avogadro or any software you can just prepare that particular drug candidate. Soft copy means you can just make a copy of that particular in this digital format, and then after you predict through many ways, and then after you synthesize, then you validate through in vitro and in vivo processes, and then after you optimize it. If the conditions are validated, then you have to scale up, because whenever we synthesize something in the lab, sometimes we prepare in millimole level, sometimes we prepare at micromole level. So that millimole, micromole is not going to get for the industry. So you have to scale up also that synthesis. That means you have to optimize that reaction conditions, and you have to furnish that for the scaling up. So if the compound is good for industrial purpose, then it is good. If it is there at lab level only, then also it is kind of a failure, even if you have been getting the very good results. So all these things must be kept in mind, and then only one should go further. So this particular loop will improve your lead quality, and this particular loop is very much adopted now in pharma R&D. So if you go for any big company, suppose Cipla, Glenmark, Ranbaxy, Dr. Reddy's, any lab if you will go, they will follow this pathway now. They have the computational bioinformatics people with them. They have organic synthetic chemists with them. They have the biology people, these people, biotechnology people who are working in this direction. So they have a team. That team will integrate to each other. The integrated team they have, and then only they work. Now, reviewed studies confirm that after following this loop, what do we get? We get faster clinical translation, improved patient safety, cost-effective development, personalized therapies. It is also possible now, case-to-case basis, to give a personalized therapy because nowadays what people are doing, some of the companies, they have a few targets in their hand, that they want to hit this disease, which is suppose 0.001% cases are only throughout the world, but still they want to solve that case. Suppose out of 1 million people, only 2 people has that disease. That kind of disease are also available, even if they are targeting those disease also. So that kind of personalized therapies also the companies are working for. What are the role of our education and capacity building? Why we are universities and academicians and other people, how they can build this kind of capacity? What we can do? First of all, we have to make our curriculum research based. Industry exposure we must give to the students, that at least within their curriculum, at least 1 month, 2 months, their summer vacation, winter vacation, whenever they get time, they go to the industry, they get that exposure, that how industries are working, so that they can just make a mindset about the curriculum. They can make a mindset about the research, that how we can do the research and how real industries are working. Student innovation projects now is the government and other people, they are also, even the companies, multinational companies and other, they have CSR responsibility, corporate social responsibility, under which they are giving so many projects. So we should go for student innovations. Faculty upskilling is also very much needed. Many times our faculties are also have been done many things in their life, but still upskilling is very much needed for research-based upskilling. Interdisciplinary learning, chemistry, biology, data science. These three must be integrated together. Data science, if we integrate with it, so chemistry and biology people can tell to the AI people that which kind of software do we need. Suppose we have some of the things in our hand. how we can improve this particular software, that can be done only via integration of these three. So team-based research culture must be there, and alignment with the sub-themes of ICHER like research methodologies, evidence-based practice, faculty capacity building, translational research, and curriculum reforms. So we as an academician, if we are having some industrialist also with us, so we should get together each other to get something concrete. But a few limitations, if you will see, yes, there are limitations, but I've already told you that many resources we have already saved. If we are saving 60 to 70% or 40%, this is a very good data. So generally, what is the problems in these in silico? Because once people are doing in silico, then synthesis, and then after the in vivo, in vitro, in vivo, and clinical trials, then the generally those who are against this process, generally those people are reporting the issues that over prediction, sometimes we get very much over predicting results in silico method. That is a problem that we address, but still it is saving too much of our resources, so we have to rely on them. Poor in vivo correlation, sometimes in silico and in vivo or in vitro results, they are not correlating to each other. Sometimes there is very much gap between these results and that results. So we think that we have wasted too much of time and all, but it's okay. Limited reproducibility. So sometimes variation in experimental protocols, differences in laboratory condition, inconsistent biological models. Sometimes it happens that we see that we were planning something else, but when we started doing it in the lab, what we were planning has been changed everything in the lab when we did it in the lab. So it happens many times. Even if it is happening, we must follow this pathway only just to save as much we can save. So regulatory and approval delays are there. We all know that whenever we want to design some drug or something in vitro, in vivo, and then after clinical trial, it's a very, very long process. Standardized protocols also sometimes we are lacking. Overall impact is, even if there are limitations, even if there are problems, we address, we acknowledge, yes, there are problems, still increases development time and cost, reduces translational success, limits data reliability. Overall impact of these limitations are this. Still there are negative impact, still we are getting something positive. That's why we are working in this direction only. So what are the research gaps? We have to identify those gaps and we have to address those gaps. So limited clinical validation, underutilization of AI and digital tools, weak academia industry collaboration, these are the problems which we are facing. Clinical trial validation is very, very limited. If we give our compound for a clinical trial, it will take a very long time to give us the answer. Underutilization of AI and digital tools, where many people are not aware about even, and they are not fully utilizing, especially in our country. Maybe in the developed country, people are using very much, but here we are not using. Weak academia and industry collaboration. Academia and industry, they are not collaborating nicely so that we can get something concrete. And sometimes in our country, we have constraints of funding and resources also. Many times, even if students are very much keen for doing research, even if the faculty members are very much keen, sometimes they are lacking funds. So if they are not having enough funds or grants, then also it is a difficult process to get the desired results. Opportunities for young researchers. Why we are conducting these kind of conferences, why we are together today for at this platform, just to create a few opportunities, just to the ideas must be exchanged, just to exchange our ideas with each other, that which kind of opportunities do we have. Like to young researchers, we can give a message that you must be engaged in interdisciplinary research. You must be developed a few skills in AI and bioinformatics. Even if you are chemistry field, even if you are any other field, you must develop. Participate in industry linked projects. Try to get, if there is a possibility, if the project is open, why not to address the projects as much as possible. Focus on clinically relevant studies. So you need to be focused on clinically relevant studies also, that how these kind of whatever you are doing can be proven as a clinical drug, can be pass the clinical trial, how it can be done. So this focus should be there from the starting. Their focus must not be only to publish the paper. Publishing paper is also a need, but with the publishing paper, what else you can do, that must be taken in mind from since inception, from very beginning. Explore startup and innovation ecosystem. Whatever ecosystems we have, they must try to get, because students are in their young age, they may work even much better, they may work even much harder and for much time, more time, even like other people are doing, the old age people are doing. So we have to address these research gaps through collaboration, digital innovation, targeted funding will empower these researchers to drive the next generation of healthcare solution. So here is a roadmap which I have prepared for a young researcher. If we have students with us, they should develop their skills within 0 to 2 years. Whatever coursework they are doing, should do that. Other skills they should develop. And then after, they should start integrating their research. Then they must go for innovation and leadership skills also. Even if they can submit their thesis, if the university or whatever institute they are working for, with that they can develop so many things. So career pathways are that we should integrate all of them: academia, industry, startups, regulatory agencies, and global research. We should see it. So then further, if we see, so emerging trends we see in this field, which are going to work too much in 2025 to 2030 are AI driven design. People are going to design too much through AI driven methods. Although whenever we are writing something, that should not be AI copied. But if there is a software which is research software and it is approved software, then that software can be AI assisted, and that is fully acceptable to the people. Digital twins, this kind of working are there in the medical field. CRI SPR based screening, that computer research integrated the screening through these methods. Nano carriers are there, multi-omics integration. So if we are these are the future trends in which people in nano field also people are working too much. Nano carriers, they will enter in our body and they will directly hit the target wherever there is a problem. Even if we have a small problem now in a body, whenever you go to a doctor, he will give you a complete list of the medicine, this 4 5 6 7 medicines, and you just end up with the fed up. Sometimes our old age parents or somebody, if we see, they just fed up of taking medicines, because we do not have target based medicines. We have so many things together. If there are disease, we give so many medicines to the people. So there must be a target based. So nano carriers can do that job. And what are the key takeaways for whatever presentation I have done today is that drug discovery has evolved into a multidisciplinary science. So this is a multidisciplinary field. We should not think that this is a single unidiscipline in which only chemistry people will work, only biology people will work. No, they have to integrate. It improves our efficiency and success rates. It remains the foundation of our innovation. Computational and biological tools, they enhance the screening. Education and capacity building are critical for sustainability, and addressing gaps requires collaboration and digital adoption. Collaboration must be there, and digital adoption is inherent. We have to adopt the digital methods which are available. We have to incorporate AI and all in these tools. Then only that can be done. So integration of chemistry, biology, and data science is the future of healthcare innovation. So data science people must be there with chemistry and biology. Earlier, the integration of chemistry and biology was working, but now the data science people are also with us. So only then these all can be happen. So in conclusion, I have already concluded in all my lecture that the conclusion is that we have to integrate, we have to keep in mind, we have to just motivate our students toward this. We have to give them an opportunity, we have to give them exposure, industry exposure, so that something concrete can be driven. And here I can show you in our lab, we are working this time in this is one pot, one step, and this is one pot, two step. Within 20 minutes, we got the products, and you see very handsome 27 compounds we got in our lab within 20 minutes of the time. And after that, first we did this screening, in silico screening of the compound, then only we designed. Now they are in the, after designing, we have been characterized them, those compounds. Now they are in the process of in vitro and in vivo. So here, like we are doing in our lab, our students, those are hardcore chemistry students, but by their own, they are doing this kind of research. You may see these all data we have been designed. So this is all about whatever I have seen. So if you have any question, I am open to them.
Okay, I think thank you, ma'am, Ankita ma'am, for elaborating each and everything, as we got to know many more things, because what we have studied, like we studied chemistry till 10th only. After that, like 12th, like those who have taken the science stream, they have studied the chemistry till 12th only. But still, like in nursing, as we can say, we won't, like we are not able to get these kind of opportunities properly until and unless we are not doing any of the research work. But in pharma department, like people, they are working with such kind of things. So thanks for this lovely session, and like we have learned a lot from this, ma'am. And I would so if anyone is having any query, they can ask, ma'am. So Sarika Saxena, ma'am, would you like to say something about Ankita Garg, ma'am? So you can proceed. Okay, no problem. So we will move on forward. So our next speaker is Sakshi Chaturvedi, ma'am. So I would like to introduce Sakshi Chaturvedi, ma'am, to everyone. So Sakshi Chaturvedi, ma'am, she's having an experience of 15 years, as she is an editorial board member of PLOS One Academic Editor, International Journal of Africa Nursing Science, Elsevier, Editage Peer Reviewer. And as if we will take as her paper published, she is having 30 paper published in Scopus, PubMed, Web Science, and Embase. As her book publications, she's having 2 book publications: Elsevier's Community Health Nursing and Book Chapters 7. And as her award, she's having 3 awards in her hand: Florence Nightingale State Award, Best Women Researcher, Best Research Paper by Rajasthan Nursing Council. And as her patent, she's having 2 patents in her hand. As her expertise, she's having expertise in modern nursing education system, community health nursing, and nursing administration. And regarding her certificate courses, she's having a certified peer reviewer, Publon, Elsevier's Ethics in Research by ICMR, and Blended Learning. As a workshops and training organizer, she has organized more than 10 workshops and training. And as a subject matter expert and as her keynote speaker, she has attended as a keynote speaker 10 of the conferences and the webinars and seminars. So, Sakshi ma'am, it's over to you.
Thank you so much, Manusha ma'am, for introducing me before the participants. Shall I present my screen or you will present?
If you want, I will do it, ma'am. I will do it.
Otherwise, I will present. No problem.
If you're willing, you can do it, ma'am.
Okay. May I know whether screen is visible?
Yes, ma'am.
So, good afternoon, one and all present over here, all the speakers, participants, and the topic which is given to me is somewhat new to nursing, but many work has been done. But we are not talking much about this topic. So I'm happy that today I have been given this opportunity to talk about this topic. My topic is nurse-led translational research, and it is an emerging field in the multidisciplinary research. What we will going to cover today is we'll know about translation research, science in nursing, what are the challenges and barriers, what are the various models available, what is the latest global development, what are the initiatives taken by Government of India, what is future vision, and what is the ecosystem for conducting translation research. So firstly, we'll discuss about what actually translation research is. Previously, when we were doing research, we were thinking that whatever we'll do, it will be used by the community. Later on, we realized that there are some problems. As such, we cannot use any bedside research. So in 1980s, the concept was research utilization, whatever research is done. Later on, we have understood that scale is also important, level is also important. Then we were focusing on evidence-based practices. Nearby 2000, we realized that if we are directly taking all the evidence-based research, it may create some issue. So we need some framework which can translate the research. And now, after 2020, we can say we are translating all those research as projects. And this particular thing is known as implementation science, and AI is the better tool while doing this translation research. So we can say translation research is a rapidly growing field of study focused on applying scientific finding to clinical practice, and it bridges the gap between research and real-world healthcare delivery. We used to say this translation research is bench to bed, then again bedside to bench. So it shows what we have done in lab, as previous speaker has explained very well about that. Whatever we have done well in the laboratory, in our research, this will be applied to clinical in the patient care setting. Then we need a framework. Then that means bedside, bench side, it will be bidirectional flow. Now, we'll try to understand what is the basic difference between basic research and translation research. When we are doing any basic research, it focuses on understanding disease mechanism and biological processes only. Whereas when we are dealing with translation research, it applies basic research finding directly to clinical practice. Basic research conducted primarily in controlled lab setting, whereas translational research is conducted in clinical and community setting with diverse populations. Means system is open. We have to control the barriers. Then only it is known as research is translated. In basic research, scientific knowledge is produced with no immediate clinical intent. Means researcher, they are not that much ready to apply that in clinical. Whereas in translational research, produce actionable evidences which are basically impacting patient outcome, and immediately it can be a policy. In basic research, result may take decades to reach to the patient. That means researcher is not sure whether it will be adopted or not. Whereas translational research, they are accelerated in nature, accelerated pathway from discovery to bedside, bench to bed, then again bench to bench. The output of basic research is, it is published in journal publication or some another models can be done, theory can be generated, whereas translational research directly lead to development of guidelines, policy change, patient care models. Like this, we can see the translational research. There are various phases of translational research. First one is T1 phase. It is known as preclinical finding. Means we are trying to do this in particular controlled setting. Then few researchers, they move to T2, translational phase 2. Community will adopt the best practice, which is practiced in lab or clinical area. That will be open for community purpose. Or sometime it may directly go to T3 phase. It can be considered as evidence, and then particular evidence will be tested and adopted. So in few researches, T2, T3, both can be there, and in few researches, one can be there. It is according to the topic of interest. T4, when the basic research, evidence-based practice, community research has reached the final stage, it will be considered to be implemented in the population. This is known as fourth phase, which is now ready for the whole population. It is now generalized. Now it is a policy. It can be country-wise, state-wise implementation. Framework in translational nursing research are, there are different type of framework according to the need of the variables. Sometimes it is theoretical domain framework, means it is related to behavior to address their cognitive factors, social factors, like this, theory domain framework. Many times we use IHI improvement model, which says plan, do, study, and then act. Another framework is knowledge to action. We create knowledge, again it is in action, then again it will create new knowledge. So this knowledge to action KTA tool, very common tool. Another very common framework is push-pull framework. It balances internal motivation and external incentive for knowledge uptake. Perry-Haas framework, very commonly used by health management study, public health workers, which promote action on research implementation in health services. Another commonly framework used is behavior change wheel. It integrates motivation, opportunity, and capability to design interventions. When we are talking about translational research, we cannot say it is related to nursing only, but it can be nurse-led. So what actually is the nature of translational research? It is 100% multidisciplinary. That means scientists from various fields, they collaborate. Nurse scientist remains principal investigator if it is nurse-led translational research. Each discipline retains its own identity. Results are pooled at conclusion of the study. Sometimes it is interdisciplinary, like yogic studies. Many times we are observing these are interdisciplinary. Cross-disciplinary problem solving. They may span nursing sciences, integration of theory across the discipline, joint methodology and analysis. Many times, translational research can be transdisciplinary. Data and procedures are shared. Scientists from various disciplines, they converge. Common goal transcend any single field, and highest level of collaboration is visible. So nurse scientists, they can collaborate with any scientist from any background to attain desired research result. This is the basic nature of translational research in nursing. When we are talking about translational research, we should know how it proceeds. So as we have told earlier also, there are 4 phases. Again, if we are talking about nursing education or we are talking about sciences, or as a postdoc person, if we are talking about. So firstly, it should be basic science, evidence generated from normal healthcare activity, the bench phase. Then clinical science, randomized clinical trial over broad population at various sites, a crucial validation step in translational science. Then it will be focusing on framework related to implementation science. That means use of identified evidence to create new guideline or large-scale practice change to improve patient outcome. At last, we will use the principle of dissemination science. That means we will involve stakeholders, policymakers. We will present that particular research so that it may be understood and gain community participation, which is the real meaning of any research. There are many challenges and barriers in translational research, like any other research. The most important challenge is evidence overload. Huge volume of research evidence make access, quality appraisal, and application overwhelming for practitioners. Then research practice gap. Many evidence-based studies remain unincorporated in healthcare policy despite of their rigorous methodology, but they are not utilized in public domain. That means they are unable to reach up to T4 phase. Methodological issue. Many times errors happen due to sample size, sample selection, other diversity, hospital and structural confound result. So they may decrease the acceptability of translational research. So that means sometimes it may reach up to T3 phase, sometimes it may reach up to T2 phase, but anyhow it will not be proceeded further, or results are not that much valid for creation of a policy. Collaboration barrier. As translational research is multidisciplinary in nature, it requires transparent communication, very clear shared goals, and clarity of roles. That means good communication is required among all translational team members. That means that particular ecosystem should be very much honest in communication, and clear delegation of authority should be there. But due to this collaboration barrier, many times researches are not reached up to T4 phase. Because of many dissatisfaction among the researchers in the team. The resource constraint. As these are long-term researches, the regressive study that usually take more than 10 years demands sustained funding, infrastructure, and stakeholder commitment. That means the stakeholder, if they are not funding properly, or stakeholder, if they are not realizing that this particular intervention is for the benefit of society, so that may decrease the motivation to do the study, and resources can be constrained. So that is the basic issue. That's why there are less translational research in many countries. Capacity building. There is need of trained nurse scientist. Nurse scientist means a person who has done PhD, then later on that person will be experienced in particular field and they will be postdoc. That nurse scientist, they are not that kind of trained to do such project. There are not defined SOPs. There is no engagement of community clearly in the intervention. Action-based researches are very few. Clinical and policy leaders, they need involvement, engagement. So strong conceptual framework, multi-center experimental design, separate experimental and control hospital, clear role definition for all team members. This can decrease the challenges related to translational research. What is the scope of translational research and career pathway? As a nurse, we should know if we have done BSc nursing, we will be research nurse, right? If we are in the research field. If we have done MSc nursing in advance, then nurse scientist like this. But if we are having a particular goal, we want to do something for a particular field. That means after your PhD and during your PhD, you should take that particular topic, work for at least 5 to 10 years on that particular topic. Then after your translation of research, that particular project will be of interest for the public domain. So postdoc is the basic thing which is required to be called as nurse scientist who can work in a transdiscipline team. And PhD nursing in India, we are not saying PhD nursing is scientist, but in many countries they used to say that PhD nursing, they can be considered as scientist. Recently, ICMR also has opened this post, nurse scientist, PhD nursing with certain criteria. So nurse scientist basically is the scope for translational research and career pathway. And areas can be related to palliative care, NCD prevention, emerging areas, pediatric and neonatal care, mental health nursing, infectious disease prevention, geriatric and genetological care, cardiovascular nursing, community health nursing. Latest global development in nurse-led translational research. There are few research projects which are openly available in public domain. In Canada, nurse-led knowledge translational program was developed in 2022. In 2023, nurse-led ICU RCT was done and music intervention was given for ICU patient on mechanical ventilation, which has significantly reduced their agitation, pain, heart rate, and now it is policy. 2024 showed NP translational framework, nurse practitioner translational framework, which is nowadays available in many countries, that nurse practitioner can be the future in clinical field. In 2024, AI and data science in nursing has emerged, and nurses led AI algorithm design, mobile health app development, data analytics for practice optimization, and representing the frontier of digital science translation. In 24-25, academic hospital partnership with $300 million investment are training new generation of nurse scientists for especially the transdisciplinary research. So this is the future of nurse-led research. Usually it takes 17 years. Till now, 32 researches are considered as translation. The nurse scientist ecosystem. How nurse scientists will work, that ecosystem we should know. We are saying that we should understand nurse-led translational research. So nurse scientist who is postdoc or advanced PhD nurse will work with community leaders, will work with policymaker, biomedical scientist, patients and families, nurse educator, hospital admin, opinion leader, and clinical researcher. So these particular ecosystem will help the nurse to translate that particular research, as she will be the principal investigator. So nurse scientists serve as principal investigator, coordinate expertise from all sectors to advance translational research towards improved patient outcome. Initiatives and way forward in India. National initiative needed. In India, there is Translation Research Center, but it is not purely related to or dedicated to nursing institution, although translational research centers are available. ICMR funded nursing grant, dedicated national grant scheme for nurse scientists under ICMR is recommended. PhD and postdoc fellowship, increased translational nursing PhD program and postdoc opportunity should be clearly developed as policy by the stakeholders, by the policymakers. Another very important thing is magnet style accreditation. When a nurse is doing translational research, it is very difficult which hospital will be experimental and which hospital will be the clinical. So in another countries, there is one thing when we used to observe translational research, it is known as magnet hospital. So adopting magnet equivalent accreditation, which will link evidence-based practice to nursing excellence. That magnet style accreditation that need to be given by the ministry or the accreditation agency. Global model, NIH, they are working for Translational Science Awards. NSTRIG network, already it is there. Knowledge Transformation Translational Challenge Program in Canada. It is highly motivating nurses to conduct nurse-led translational research at multi-site. And magnet designation hospitals are already given in many countries for the conduction of translational research. Translational research is very much useful and implemented in nursing. Why we are saying so? It is basically important for community health nursing. It is also important for medical, surgical, pediatric, mental health, and other things. But until unless the knowledge of the translational research is not known to community nurse, it cannot be translated. So what are the key roles of community nurses? They should be uniquely positioned for leadership in community-based research network. Foster collaboration among stakeholders, including older adults and vulnerable population. Bridge the gap in care continuum and care setting transition. Extend patient and family advocacy into research arenas. Lead community engagement as core of translational initiatives. The latest intervention which we have seen as translated, which is under translation, is coconut massage oil, which was practiced by nurses. It was nurse-led research. Many researches you will see that if nurses will give coconut massage oil therapy to premature newborn, it will help in improvement of weight gain. So now this particular research, that means, will be adopted by the nurses who are giving health education to community members if they will practice so. So community health nursing is the important field for that. And when they are adopting this, intervening this, then only it can be translated. Still, it is under process. Although there are many evidences available, still people are facing this problem of birth weight management in premature newborns. So community research benefit will be, it will address rural health disparity, engage patient in all research phases, inform clinical practice with direct implications, and apply cost-effective prevention and treatment according to the need of the community. The researches will be done, like in China and other countries. Future vision for 2030, the nurse-led translational research are increasing, and AI and machine learning models are available to identify translation opportunity from electronic health record and genomic data. And health, mobile health, and digital trials are available. Precision nursing as per the genomic and biomarker guided nursing intervention according to the need of the individual. So these are the few emerging areas where nurse-led research can be done. Global Research Network, cross-continental nurse scientist collaboration model on NSTRIG and CTSA consortia. Sustainability and health, translational research address climate health intersection and sustainable care model. By 2030, nurse scientists are projected to lead over 600 indexed translational publication annually. So it is projected data. Thank you so much. And if you want more information regarding translational research, we have written one article in 2022, Nurse-led translational research, an emerging field in multidisciplinary research. You can go through that and cite this. So much, Manisha ma'am, for sharing this wonderful opportunity. Now, I would like to take any question if it is there among audience.
Hello, madam. Good morning, madam. Hello.
Good morning, Aarti ma'am.
Yeah, madam. This translational research, how much time we required for this study?
Ma'am,
Is
previously
there
it
any
was nearby.
duration? Yeah.
Previously it was nearby 10 years, but now due to AI help and other things, technology is available. Many translational research has been enhanced within 2 years or 3 years. They are available for policy.
Okay, ma'am. Thank you.
Thank you, ma'am.
So we will move on to the next speaker, and the next speaker is our Ruchika Rani ma'am. She is assistant professor in Rishikesh. As an education and training, she is graduated from Christian Medical College and Hospital, College of Nursing, Ludhiana, Punjab in 2004. She has completed her post-graduation in oncology nursing from Maharishi Markandeshwar College of Nursing, Mulana, Ambala in 2009. And she has completed her PhD from INC, Indian Nursing Council in collaboration with Rajiv Gandhi University of Health Science, Bangalore in 2023. And her PhD topic was a randomized controlled trial to assess the impact of a nurse-led pre-chemotherapy education program on quality of life, symptom experiences, hospital visit, and psychological distress among breast cancer patients in selected hospital in Uttarakhand. As a teaching and as administration experience, she worked as an assistant professor, College of Nursing, All India Institute of Medical Science, AIMS, Rishikesh, Uttarakhand, India, from 13 August 2018 till date. And as her previous experience, she was a clinical instructor at Shahid Kartar Singh Sarabha College of Nursing, Sarabha, Ludhiana, from 1st August 2008 to 31st July 2009. And as a research interest area, breast cancer, head and neck cancer, quality of life, body image among cancer patient, chemotherapy with radiation therapy, nursing education students, clinical nursing skills and patient care and mentorship. So it's over to you, ma'am, and I will share your slide so that you can present your topic over here. Ruchika Rani, ma'am, are you there? Ma'am, are you there? Are you available?
PPTR, no, so ma'am.
Okay, wait.
Visible, ma'am.
Okay. Ruchika Rani, ma'am, are you available?
Hello.
Yes,
Am I
ma'am.
audible?
Yes, ma'am, you are audible now.
Okay, okay. Thank you so much for the nice words. And Dr. Manisha, for your continued support and in touch and everything you have given me, whatever we have supposed to you. And thank you so much for giving me this opportunity to be the part of this DIAR Foundation and this conference. So can you put it in the slideshow?
Yes, ma'am. Are you able to see, ma'am?
Yeah, I can see.
Yes, ma'am.
Okay. So today's the topic for discussion, that is competency-based education in nursing. So I can understand that the most of our presenters or most of our like the delegates, those who are present here, they are from the nursing sides. So here the importance of educational competencies. So how it has been changed. So first of all, I would like to say that as there is so much advancement in the medical technology, so we are having good AI methods, there's diagnostic improvement. So we are having the more aging population and more need of nurses there. We need the quality nurses, and the nurses, those who don't require only knowledge, they are not packed with the knowledge, but actually we want nurses, those who are competent enough to provide the care to this population who require these services. And so these gap the gap from the challenge to practice, that can be only completed when we are having the nurses, those who are actually competent enough to provide this type of care. So for this, if we can see the future of the nurses, now we have to change the academic pattern also. Now the academic pattern, that we have to shift from the traditional pattern to the competency-based pattern. So like in the traditional pattern, we are saying that more emphasis is put in the content delivery. We are just focusing on the content part. We want imparting the knowledge to them and cognitive part. But now we would like to have that the nurses are not only competent in their knowledge, but also they should have enough skills. So they are providing the services, they're providing the care with good practice, with quality services also. Even in the, if we can see that there is one evidence that shows that the Institute of Medicine, where it has been said to err is human, that report, that is very much common in the clinical areas. There are so many like medication errors or incident reports used to occur in the hospitals. So they have highlighted that now there is a critical need for the nursing education that address the quality and safety for the patient care. Rather than just we are producing the nurses, those who are packed with knowledge, but they are not competent enough to provide the care. Next slide, please. So here the outcomes, but the objectives we have planned for this today's session is that first we will want to see what is the definition of this competency-based education, how to assess, how to analyze, what are the various methods to assess, how to implement, what could be the outcomes of such competency-based education, and what we can see in the future. Next, please. So here I would like to put emphasis on that how the nursing education actually has evolved from the 1860s to today's time. If we can see that from the previous Nightingale's era, that here the hospital-based apprenticeship models were used, where the main focus was just to complete their task rather than putting the theoretical knowledge. But with the passage of time and with span of time, now the nurses are they were packed with their knowledge. Their theoretical foundations were putting strongly, so that the more focus was just put on the content delivery. And till now, we are again, there was some improvement that nurses have started to put more emphasis on the problem-based learning. So like we can also see in their multiple choice questions, we are preparing the nurses that how they are critically they are thinking for these things. But the thing is, we are having nurses, they can think critically, but now the more focus that should be put on their skills. That if the nurses are in the clinicals, they are good with their knowledge, but when the patient require care, when the patient require that quality care, here the importance of competency is actually coming. So we don't want the nurses they are having their certificates, nurses they have completed their credit hours, but they are not competent enough. So that is the actually basis for this foundation of this competent-based learning. So this is actually a teaching learning approach that enable the students to develop and grow based on their ability to learn or master skills. The focus of this competency-based learning is actually we want our students to get mastery over the skill at their own pace. That is very good part of this competency-based learning. That is, we are actually preparing or mastering them on particular skill, but their own pace. Here we are not time bounded. The main component of this is that it is not time bounded. Like in our programs, if we'll see that in the syllabus or in our curriculums, we are packed with that particular limit of time, that we are not focusing if our students are competent enough, or graduates now are moving outside to the clinical areas or hospitals, they're handling the patients. Are they competent? Are they having appropriate skills so they can manage the patients? But here the more focus is on the mastering the skills at their own pace, because it is actually individualized approach. It is not the approach that is usually the traditional class-based approach. So we can see that the main components of this CBE is that competency achievement, that is mastering the skills, and focus is more on the performance rather than the time spent. The students are again and again, we are sending them for the to get the skill or to get the mastery of the skill. Until they are not master of their particular skills, they have to again give that test, or they have to undergo the same performance again. So that's why that is the main focus is on the performance. We are not focusing on the time spent. So competency, it is a packed set of the knowledge, skill, and attitude. So we are previously we are focusing on the knowledge, then in between only the skill, and again now it is a complete package of knowledge, skill, and attitude. Attitude also implies the communication part, how the nurses are communicating, how they are delivering the health education part, and how they are empathetic towards their patients. Next, please. So this is the comparison between the traditional and competency-based education. How we can see that how it is different from the previous what we are teaching our students. So if you see the design of the traditional, it is content driven. So according to the Indian Nursing Council or the any other, it's in other specialty areas, we will see that we are having a lot of content, and it is according to the discipline based. It could be the medicine, it could be paramedical, whatever. But here the focus in CBE is only on the competency. If I can give the example of this. Now again syllabus has been changed by the Indian Nursing Council also. New modules has been added. If you can see that in each semester, the student have to complete one module, like first aid module, they have to complete the palliative care module. So these are the modules actually based on the competency. So we are giving the students that demonstrations, and we are again assessing them through their OSCEs or multiple choice questions. We are just thinking it is also outcome focused. Are the students are actually trained, they are skilled enough to give that particular procedure. Similarly, in the traditional way, the learning approach is very passive. It is only for the lecture based and fixed time. We are having particular hours are given, and semester hours are there, and based on the knowledge recall. If you see that our test, the evaluations are considered only on the basis of what the cognitive part or cognitive knowledge they are having. And students are also in the passive form. They are just progressing uniformly. The focus is not only particular student to the whole class. And teachers' role in the traditional part is only to transmit the knowledge. They are just content expert. They are giving the knowledge, and they have to achieve their complete their course, and the evaluation is done usually at the end of the course. But now, if you see in the CBE, here the focus is on the student. We just would like to know what is the nature of the student, but what are the shortcomings of the student, why the students are not achieving the competency. So with the process of OSCEs, with the MCQs, we are assessing the knowledge, we are assessing the skills and competency of the students, and teachers are taking the part of facilitators. They're just facilitating, and they're assessing the competency of the student, that the student has mastered over the skill, and they're ready for the practice. And evaluation part is formative and continuous. It is not like that it will be evaluated only at the end of the session, but it is with the debriefing and with this continuous feedback, the student knows that how much knowledge or how much skills they have incorporated. Next. So this is how the shift has been now. The key has been shifted, from the tradition, from the points and percentage. Previously, the students just focusing and they have to get the grades, they have to see their percentage, how many percentage. Now the focus is on the feedback. Their facilitators, their teachers, their mentors, what feedback they are giving, actually, it is based on that. If the student has achieved particular standards of that particular skill, now then it has been achieved. Similarly, the content from the discrete content and standard to the whole learner outcomes. Similarly, from the seat time to, rather than spending, like student have to spend enough time in the classrooms and they're sitting for a particular time, here the focus is on the efficiency. Until the time they are efficient in their skills, we have to focus on that. So I think we have understood what is competency-based education. It is that like learners advance upon demonstrated mastery of the clearly defined competency. The focus is the competencies are clearly defined according to the standard criteria and integrated. 3 components are integrated, that is knowledge, skills, and attitudes, rather than just accumulating the credit hours or seat time. That how, like if I have spent 30 hours, now I will be like, getting this particular certificate. It is not like that. Main thing is, if I have achieved that mastery over the skill, then I can only get that particular certificate. So these are the core principles of CBE. Like first is clearly competency standards are there. There should be authentic assessment by the demonstrations and personalized learning pathways, and continuous feedbacks are there through the formative assessments. Next, please. So this is the Bloom's taxonomy. This is this pyramid, or we can say this is the hierarchy given by the Benjamin Bloom in 1956. So they have focused that if we see the largest or higher part of this, that is to create. Now, this is the larger hierarchy, right? So that shows that if you want to create, it is the higher level of hierarchy is actually dependent on the lower level of hierarchy. The first part is remember. So if we'll see that in the student life also, the focus first, in the first year, we are just teaching them the anatomy and physiology and such type of subjects. The focus is only just to remember, that they should need to remember what are the anatomy of the respiratory system. And then they have to understand how this, that is the physiology, how it is actually happening in the body. And then they learn to apply the principles, like as how the breathing happens in the body, how the, what is the asthma type of conditions are there. Then they analyze the different, they compare the normal and abnormal with sounds, and then they evaluate what will happen. And similarly in the create part, they can do the many researches, what type of breathing techniques it can be helpful to improve the health of the patients. So these are the Bloom's taxonomy that shows that everything that if we want to achieve the higher levels of this Bloom's taxonomy, first we have to come to the lower levels. They are all integrated parts. First we have to remember, then understand, apply, analyze, evaluate, and create. Similar principles are used in the CBE. Here we actually, it is not only that we are just focusing on the skills. The main part is, first of all, we let the students to remember what is actually the basic recalls, basic memorizing the things, and then we tell them to understand and then apply and then learn the higher levels of Bloom's taxonomy. Next is. So this is the Miller's pyramid for assessing the clinical competence. Like in the previously, I have shown the Bloom's taxonomy. It is for the, like how the knowledge is regained, how the knowledge is achieved. Here we want to see how the skills are achieved. See, whatever we do, first of all, we get the knowledge and then we learn the skills. Similarly, if you can see the orange part or the broad part, that is know. First, we know what is how much I know about that particular procedure, particular thing. And then I would learn knows how, right? Then we learn how to clinically apply that thing, like through the case presentation, through essays, through MCQs, and then comes to the, I know and know how is that is the application part, then we demonstrate, that is shows how, right? It is a demonstration of learning. We teach the student through the simulations and all skills. And last part is does. The student or the performer is now performing independently because that person has learned, that person has gained the knowledge through the that learning part and then understood the skills that through the case presentations and simulations. And now that person knows how to perform that practice through the direct observation or workplace based. So these through 4 things we can see the knowledge, skills, and attitude. You can see that there's a novice, then the person becomes expert. So these are the core components of the competency-based education. So these are the 5 components when we are thinking for the competency. That is, there should be competency that should be clearly defined, that there should be authentic assessment way and personalized learning strategies. That is, we consider each individual is unique and we want remediation. Even if that person has not achieved, then remediation opportunities are given. There's a robust feedback mechanisms are there, continuous quality improvement through the formative assessments are there. Next, please. So these are the domains, if we'll talk about the nursing competencies, where we want our nurses to have mastery over their knowledge part, the person-centered care, knowledge about the population health, epidemiological principles, community knowledge, evidence-based nursing practice about the research, methodologies, quality and safety principles, error prevention strategies, interprofessional partnerships, system-based practice, how to get the resources, how to make the policy implications, informatic and healthcare technology. They should get abreast with the recent technologies and knowledge, AI technologies, professionalism, ethical components, accountability part, personal, professional, and leadership development. These are the 10 components or 10 domains the nursing competency should be developed. So first part is clinical skill competency. So here the focus is to make the nursing student to make the expert, because as they're entering the profession or as they are entering to this program, they're just very novice to each and everything. So now the main objective of any curriculum is to make them novice to expert. And this is skill acquisition framework. So when they're novice, we can say that we are governing their behavior, we are guided through their protocols and procedures, and then they are competent, becoming the competent practitioner, where the main goal is how to efficiently organize their efficient organization of actions, how they are doing, how the skills are performed. And last one is when they're getting expert and get proficiency in their clinicals. So it is the holistic understanding of the patient's needs. So when if the talk about the principles, so these are the goals should be clear. It should be focused attention should be given to each and every student, immediate feedback like real-time coaching and correction, and then remediations and repetitions were given to that particular skill. And then we can see that the progressive improvement in their skills. And how these competency can be validated through the skill checklist, demonstrations, return demonstrations, or skills. So these are the ways we can see their clinical evaluations, and these are the ways of how we can evaluate their competencies. Next, please. So communication and professionalism competencies. So these are the 4 components where we'll talk about the communication and professionalism, like therapeutic communication, interprofessional collaboration, cultural competency, and professional identity and ethics. In the therapeutic communication, the students we are making them competent, how to do the active listening, how to give the empathetic response, clear information, how to resolve the conflicts. Similarly, when they're doing any collaborative work with the interprofessional department, they should have clarity about their roles and about the complementary contributions in that particular work, team communication, shared decision making, and mutual respect. So similarly, cultural competency. So when providing the care to the patients, are the students aware about their culture? So cultural knowledge, cultural skills, health literacy, that part should be incorporated. Similarly, professional ethics, ethical decision making, accountability, advocacy, that make the student lifelong learner. And even these things can be also assessed through the skills communication part also. Then moving to the critical thinking, how we'll see the critical thinking in the clinical judgment. So it can be seen through the clinical scenarios also. Even we can put the MCQs in that scenario parts also. So we can judge how much critical thinking they have developed. So in the advanced, like when they are just not novice, now they have learned how to apply the knowledge. In that part, we can help them to understand the case studies, concept mapping, deep questioning, reflective practice. Like in the, if you can see that, like in the BLS, when we are teaching the BLS and in the demonstration. So we tell the student first we are demonstrating the actual part, all the steps of the BLS, and then we let the students to do on the dummies. So when they're practicing, if the student has done anything wrong, then the reflective practice that should be there, where the students can be asked why they have chosen this first step. Like if the student, if it's saying that, "Okay, first of all, I will do the airway management." So we can ask the student why only you are focusing on the airway. It is why it is not the compression. So that is called the reflective practices, where the debriefing sessions and all the things can be. So the student may know that the critical thinking part can be enhanced in them. So these can be assessed through the strategies like NCLEX, simulation debriefing, case studies, OSCE scenarios, and peer reviews. Next, please. The fourth component is patient safety and quality care competencies. So here the focus should be already as told that this QSEN training, that is quality, safety, and educating nurses. So they have actually focused on more part on the safety of the patients and quality care, whatever that's why this CBE actually emerged. They have thought about that we want nurses, those who provide care with quality and taking the patient safety in their mind also. So in this safety science, like the person know human factors engineering, what are the human limitations, like errors could be from anyone, and like humans to err is human, so anyone can do these mistakes. So here the thinking is that we should make the persons being accountable for that error also, and there should not be no blaming approach should be there. Like if the error is happening, we should go deeply through that part. It could be through the administrative policies, it could be through any mistakes from the system side. So that part should be, analysis should be done. So we should more focus on the error prevention strategies rather than making the person blaming on that. So medication safety, infection prevention, fall prevention, communication safety, equipment safety, these are the things where the students can be competent enough how to prevent these things. So quality improvement competency, these are some cycles or analysis are there. So evidence of CBE, it has shown that when they put the CBE in the practice, it has actually improved the patient safety, and the nursing also have improved in their attitudes, decreased medication errors, and it is also preparing the graduates for the error reductions. So this shows the role of simulation in competency-based education. So when we are talking about that we should prepare the students for the mastery over the skill, that means first of all, if they are novice, then we have to make the students to practice on the dummies or in the simulators. So nowadays simulators are not like a very traditional dummies only. They are actually creating the real life scenarios are there. So we are having different type of simulator, low fidelity, medium, high fidelity, and even the standardized patients are also used. So the students know before moving to the clinical areas, they know that how to handle the patients, how to do the procedures. So it create the safe environment for the student as well as for the patients and prevent the harms to the patient. And repetitive practice, it gives the mastery over the skill to the students. And similarly, standardized experience with the standardized patients or persons, it helps the students how to do the communication part. So they learn, and even they get the feedbacks, immediate feedbacks, and it helps to develop the confidence among them. So debriefing, it is the most critical component of it. It actually debriefing with good judgment, where the main things we are just observing the reaction of the student, we are analyzing and then summarizing what the student has done, what are the wrong things he has performed, and where the chances, where the things we have to improve. So it will more develop the clinical reasoning among them. So this is how after the simulation, used to have debriefing sessions. So OSCE, OSCE, you all know that this is the standardized assessment methods where the candidates rotate through various time stations and they demonstrate clinical competencies on the simulated scenarios. So the stations could be from like various stations can be developed, like it could be history taking or physical examination, various procedural skills, data interpretations like lab test or ECG. So there can be various stations can be developed, or generally we used to arrange 5 to 10 stations for, and 5 to 10 minutes are given per station. Standardized patients can also be used. Trained examiners should know how to get the OSCEs and simulations. So these things are required for the OSCEs. Next. So that shows a technological integration in competency-based education. So nowadays, like from traditional part, if we'll say that the content is delivered only through the education part. Now we are focusing on how we can integrate the technology into the competency-based education. Like, if I know about my student, I have given the demonstration, but I don't know actually he or she has developed that particular mastery over that procedure. So this competency tracking is a way of this technology that we can track on the simulated patient if the student has done any mistake, that competency trackers, they will note down through the feedbacks or through the you can say checklist, they know that the student has performed this thing wrong. So for the next time, we just focus on that part also, that students are students are getting prepared enough to get the mastery over that part. So this is curriculum mapping, that we know that the students are actually from the novice to now they are learning, becoming expert. Similarly, there are various tools are there, skill labs, video libraries, peer review platform, digital badging, AI tutoring, where the students are performing on the simulators. They are learning that how much efficiency they have achieved and how much improvement they want. Their virtual tutors are also there in that. They are teaching them that how much improvement they have achieved or how much things to be improved further. Next, please. So these are the e-portfolios and continuous assessment. So as like in the previous, I'm saying that we have to go through the that we can say that summative evaluation. That is at the end of the, we are just seeing their question that exams, and we are giving them grades and points that how much they have achieved. But that is only on the focus is on the cognitive part, but not on their practical part. Practical part is also there through the OSCEs, but that is not enough standardized way of arrangement is there. So e-portfolio systems, like it is the digital repositories. They are documenting the students' competency development across the nursing curriculum through the formative and summative assessment. So they know how much level they have achieved. They are on the level 1, 2, or 3. Level 1 means direct supervision is required, like for the first year student. Level 2 is indirect supervision is required. Now they have improved enough. But level 3 now, like we are producing the nurses at, like they are the interns, they don't require any supervision. Now they are prepared enough to become the independent RMs also. So these assessment we are doing longitudinally, and multiple data points are getting self-reflections, and even the early interventions. If there is any mistake, the students knows how to improve themselves. So there are various methods of assessment: direct observation, mini-sex, case-based scenarios, 360-degree feedback, and reflective narratives. So these are the things which helps to produce the e-portfolio of the students. So these are the benefits of competency-based education in nursing. If I'll talk about for the students' benefit, they have clear expectation. They know what I want to achieve through the 4-year programs of the nursing or any programs. So they are progressing at their own pace with the remediation support. They know where I am wrong. Until I'm getting this mastery over particular skill or particular content, I will not get the certificate. So he will get self-efficacy, will get the confidence among them. And of course, we are producing the nurses with more capabilities also. For the faculties also, like they know what to teach. Their focused teaching is their objective assessment part is there. Now the roles are getting changed. Now they are not only the content delivered, they are actually facilitators. They are helping the students to achieve the competency. And healthcare system, of course, we are having the nurses, those who are practice ready graduates are there, they are competent enough. We are more sure about the patient safety, quality care of the nursing care, and workforce retention was there. So this improves the satisfaction among the employers also. Next, please. So this has shown how much, there is a research study that shown how much improvement in the patient outcomes can be seen through the quality of care, clinical judgment, this medication errors, how these things have reduced with the help of this, producing the competency-based education. Next, please. So already I have told about how the role of faculty has now been transformed from the content expert to the just facilitator. They are not lecturers now, they are coaching. They're giving the coaching to the students and competency assessors and curriculum designers. So these are the things we have already discussed about that. Next, please. So student perspective, already we done. So they are having the clear expectation. Now the challenges, what challenges we can see. Next, please. What challenges could be found through the faculty, student related and resource infrastructure related. So resistance to change. Faculty, actually, they don't want to change themselves to the new ways of teaching. So it is very difficult for them to remove that old strategies, and because it has of course it has increased the workload, because we have to make the syllabus in such a form that we divide the syllabus into a competency forms. We have to see how to get the competency among students. So various assessment skills, technology, we have to get like this, what the new technologies are coming, we have to see, and we have to understand their ways also. So it is sometimes difficult for the faculties also. For the students, again, the same part is they have to sometimes they feel difficulty in adapting to the new learning models. It creates anxiety among them when they are doing the performing in OSCEs also, simulations. If they are not getting mastery over them, it may create anxiety among them. Or sometimes some students need more time to get the mastery, so they feel some lacking part that. And peer competition could be there. Similarly, infrastructurally, if I'll say that there is also some challenge. Financial investment has to be done, physical space requirement is increasing, personal needs and technology infrastructure, we need all the things, then we can only perform these competency-based things. So what could be the solution? Solution is on the basis of the phased implementation research. We cannot just put all the things in the first phase only. We have to see in the short duration and of the for the small number of groups. So faculty development programs, curriculum programs, there are a lot of curriculum programs, and even key faculty development is also going through in our country also. So that has to be expanded. This implementation has to be expanded throughout the country, and this faculty development strategies, it should be workshops, comprehensive workshops, and conferences should be there to get that. And similarly for the stakeholders also, we have to get the administrative support, accreditation bodies, and through the state boards, we can see the smooth running of these CBE programs. Similarly, technologies also. Next, please. So these are the future directions in competency-based nursing. So this is the future of all the programs, specifically for the healthcare professions. Now we have to come out of through these books, and we have to go outside. It is not only the content or knowledge we know, because in this era, we are already packed with the lot of content, lot of knowledge around us. So much knowledge is already there, but actually we need the skills and mastery over them, how to apply this knowledge. So these are the futures. So we have to get pack up with all these AI-powered learning, immersive technologies, and micro-credentials things. Next, please. These are the immersive technologies. Please, last slide, please. No, that in the with the images. So these are the immersive technologies with the AI technology. So here you can see that the person has put this VR goggle, so they know how to see. They're not just only learning, they know if they're doing it properly, what will happen. If they are not doing it correctly, then how they can lose the patients. 3D models are available. So the students know it is not only basically, it is to enhance their all the levels of the Bloom's taxonomy, if what we have already done. Next, please. So, key takeaways, CBE is actually focusing on the mastery, not the time. So, they have already American Association, they have provides a framework in the India also. We are using it through the this in the MBBS curriculum also, even small limits in this nursing curriculum also. Simulation and OSCEs are the critical tools. We require systematic approach. Faculty development part is very essential, that we have to remove the traditional approach, and it enables the CBE at the scale levels. And then there are so many challenges could be there, but we have to overcome all these things. Next, please. So in the last slide, so it represents a fundamental paradigm shift in the nursing education. So we are preparing the nurses with practices, knowledge, and attitude, and safe and effective patient care. So it improves clinical judgment, patient safety. It creates confidence among the graduates and meet the complex needs of the contemporary healthcare. So these are the references what I have found. So any question or anything want to add? No questions means, have you understood, or it is not like?
I think everyone has understood very well the topics, ma'am.
Thank you so
Thank
much.
you, Ruchika, ma'am, for this wonderful presentation.
Thank you so much.
You're most welcome, ma'am. So we will move forward to the another presenter, and the presenter, our spokesperson is Srivani, madam. Ma'am, she is available there. I think, let me check. She is not available right now. By the time I would like to. Srivani, ma'am, she's.
Yeah, I'm there.
Okay, ma'am, you are there. Okay. Nice to have you on board with us, ma'am.
Yeah, can you give the access for sharing?
Sure, ma'am. We will do it. So before starting up the ma'am's session, I would like to introduce our Dr. Srivani R. Ma'am. She is a professor and HOD in Dharwad Institute of Medical Health and Neuroscience, Karnataka. She has completed her PhD under National Consortium for PhD in Nursing, Rajiv Gandhi University of Health Science, Karnataka in 2013. And she has undergone Body, Mind, Spirit Practitioner course at Hong Kong University, Hong Kong in 2011. And as her sponsored research project, she is a principal investigator for she has completed 4 research projects under ICSSR, Ministry of Health and Family Welfare, Government of India, and Hong Kong University. So let me tell one thing more about Srivani ma'am, because when we were in college time, we studied ma'am's book. Still we refer ma'am's book, because there are like lots of things are there. Because sometimes what exactly happens when we read some author's book, or like if we are getting connected to someone, so definitely the things are there. And today also we will learn many more things from ma'am's. So ma'am, it's over to you. Ma'am, did you get the access?
1 second.
Yes, ma'am.
Yeah, I'm trying to share.
Yes, ma'am.
Entire screen. No, no. Let me try.
Yeah, ma'am, you just try it once.
Yeah. What I should pray that press show my screen anyway, or
No, ma'am, you have to go back. You have to go to the PPT, and then
yeah, I have done actually.
and for scrolling it down, ma'am, there is an arrow button. Just
Yes,
check
it,
it on
it
the
is
right
not
hand side. Yes,
moving.
ma'am.
It is not moving. Okay, okay. One more time I will share.
Yeah, you just try it once, ma'am.
Is it like a window I have to share or
Ma'am, you have to share the window.
window only?
You just
no?
check it. Yes, ma'am.
The slides are not moving. That is the problem.
Shall I do it?
But the thing, no, many times you have to press. You should have a lot of focus because many slides are there. I made animations also. That is why.
Okay, I will do it, ma'am. Don't
Okay.
worry.
Then. Okay. Okay. Then.
I'll do it, ma'am.
Okay.
Now it's fine, ma'am.
No,
It's
not-
visible.
No, no, not visible.
Not visible till now.
No, no.
Okay, okay, ma'am. I'm checking it.
It is there properly. Don't know what is the problem.
Ma'am, now
No, it. No, no. It is not visible. Only this screen only visible.
okay, ma'am. Wait, I will do it once again.
Yeah, it may come now.
One second. I'm doing it. Why it's showing pause from my end?
Wait, I too will try.
Yes, ma'am. I'm doing it, ma'am.
See, it has come, but it is not moving again.
From your end, ma'am.
See, it is not moving. Slides are not moving, not going to presentation mode. It is in the slide mode only.
Ma'am, kindly start the slideshow by clicking on the menu bar slideshow.
Okay, okay, 1 second then. Okay. Share. Yes. Yes, it has come now. Where I should go? Menu bar.
Yeah, slideshow.
Slideshow.
On the top, you can see home, insert, design, animation, slideshow.
No, it is only screen is visible because I am unable to operate this one.
Ma'am, when you are sharing the screen, that time you have to select entire screen. not
Entire
only
screen.
window.
Okay, okay, okay. Yeah, in the entire screen, that PPT is not visible.
When you will share it, then it will come.
Okay, okay. See, it is coming.
Now, ma'am, open the PPT.
Yeah, I opened in my this one desktop. It is opened.
From the down menu bar, you have to click on the PPT.
Okay.
You have to switch from the Chrome to Microsoft PowerPoint.
Okay, Chrome 2. Okay, so entire screen I have to press.
Yes.
Then after that,
Down where the PowerPoint presentation is there, you have to click on that from the taskbar.
it is coming like that only.
A bit down, down.
Yeah, down. Yeah.
The
Down.
first one.
Ah.
Yeah, click here. Now
Yes.
press F5. Now click on the slideshow over the top.
Oh, okay. Yes.
Now from beginning.
From beginning. Yes.
Yes, ma'am. Your slide is visible to all. Everything is working smooth. You can continue the
No,
session.
but Yeah, it is moving also. Okay. No, but it is not moving. Yeah, with the only click the button, it is going. Okay.
Yes,
Is it
ma'am.
okay then? I'll go
Yes,
ahead.
ma'am, it is okay. It is okay. You can use the left and right arrows to navigate.
Okay, left and right. It's not coming from the first. From the beginning, it is not coming.
Ma'am, and show.
Huh?
Just click on hide first.
Hide first. Okay. Hide. Yes.
Yeah. Now press escape button from the keyboard.
Yes. It's not going.
Ma'am, just give a right click on the screen.
Yes. Right click. Previous slide. Shall I go?
Yeah, previous. More previous.
I think it may go.
Same way.
Yes.
Yeah,
Yes.
Yes. Once more we have to do.
Yes.
Yes, ma'am. Now one.
Is it okay? Shall I
Yes,
continue?
ma'am. Yes,
Okay.
ma'am. Continue.
Manisha, is it okay? Then I'll start.
Yes, ma'am, you can continue.
Yes. Yeah, dear audience, happy afternoon to one and all. I am thankful for Manisha for giving an introduction about me. Let me start my program with the experimental research designs. The session objectives. After completion of this session, you will be knowing the definition of nursing research and various types of experimental designs in that true experimental and quasi-experimental. What are all the key features of each design? The strength and limitations of both experimental and quasi-experimental designs. Yeah. Let me tell you, the quantitative research designs are classified as 4 types. So basically the research designs are quantitative, qualitative and mixed methods. But today we are focusing only on the quantitative research, in that also only experimental research designs. Okay. Before coming to my topic, I want to tell the basic types. In the quantitative research, we have a non-experimental designs and experimental designs. Under the non-experimental designs, you have a descriptive and correlational designs. Under the experimental designs, you have a quasi-experimental and experimental designs. So these are all the 4 basic classification. But now we will move on to the experimental research design. What is experimental research design? It is a study method where the researcher deliberately introduces an intervention. Means purposefully he gives the treatment and control the conditions to see its effect on the participants. So that is the main thing. This design researcher typically what he does, he will manipulate something. Manipulation means giving a new nursing intervention, use controls and experimental group, both control and experimental group. He will randomly assign the participants to the control and experimental. He will measure the outcomes objectively. So here, what is experimental design means, here the researcher deliberately gives the treatment or introduces the intervention, controls the condition to see the effect on the participants. Basically, what he will do, one is manipulation. What is manipulation? Maybe giving a additional intervention or modifying the existing intervention. Sometimes it can be a deletion of intervention also. Any 3 things can be done, comes under the manipulation. He will use both control and experimental group. Then assign the participant randomly to experimental and control group, and he will measure the outcomes objectively. These are the 4 components of experimental design. Then why we need experimental designs? Always experimental designs will provide quality evidence for the clinical practice. What is that quality evidence? Quality evidence means whenever we conduct experimental designs, the outcome will give a quality evidence. Means the outcome, we have a faith on that, and this outcome will add new knowledge to our nursing interventions, provide justification for interventions. Every day we are doing some nursing interventions to patients. Why we are doing that intervention? By doing this one, what the patient will have improvement. So that justification will come through experimental studies. For example, if the person has a back sore, bed sore, then you will be giving a back massage. So through the studies, we'll come to know with the back massage how much improvement will be there. how frequently we have to do back massage, all the justifications we will get through experimental designs, and it improves the patient outcome and the quality of life. Organizations get accredited status. Accredited status means NAT accreditation, NABH accreditation, all those things. One criteria is there that to particular organization should conduct experimental studies. These are all the more important reasons for conducting experimental designs. Now we will move on to the types of experimental designs. I already told that experimental designs are true experimental and quasi-experimental designs. Most of our nursing textbooks, this classification is given, but whereas in other books, this classification is not given. So all comes under the experimental designs only. Now the first one is the true experimental design. True experimental design is a study where the researcher introduces an intervention, controls the variables, randomization. There are three important things are there in experimental design. When we call it as a experimental design, the person means the researcher introduces the intervention that is called as manipulation. He will also controls the variable by having experimental and control group, and he randomly assign the subjects to the both experimental and control group. What is manipulation? Manipulation means deliberately introducing an intervention or a treatment. Control means controlling the extraneous variable, conducting the study in a controlled manner, in a laboratory manner, more structured way, more focused way. We also can have a control by having the control group and experimental group. Randomization means participants are randomly random. Randomly means without having any bias. Equally, both experimental control group participants will get the subjects. That is the meaning. Am I audible? Is it okay?
Yes, ma'am.
Yes, you are audible.
Okay, thank you. Thank you. So now we will see what are all the symbols are used in the experimental designs, because many times you will see R, O, X like that. So I thought let me explain those things. R means random assignment of subjects to the group. O means observation or measurement of dependent variable. Experimental treatment, X means experimental treatment or intervention. So these are all the symbols you remember, because sometimes, you know, with the symbolically R, O, X, they will be giving. So for that, I have clarified here. Now we will see the types of true experimental design: post-test only control group design, pre-test, post-test control group design, Solomon 4 group design, crossover design, factorial design, and randomized block design. There are the 6 types are there. In detail about these types, I will be explaining. The first one is the post-test only control group design. The other name for this is after only design. So here what the assessment will be conducted after the treatment. When you see this picture, you can see the subjects are randomly assigned to experimental and control group. A treatment is given for the experimental group. Control group may receive the routine treatment or no intervention. Post-test is conducted after the intervention. Now we will see dependent variable is measured only once after the experimental treatment has been administered. That is called as post-test only control group design. Now we will see the steps. Randomly assigning the subject to experimental and control group. Subjects are not pre-tested. Experimental treatment to subjects in the experimental group. Routine treatment or no treatment to the control group. And post-test is conducted to everyone. Coming to the example. Here, in which conditions we can do that also I will explain in the later slides. The example is a study to evaluate the effectiveness of antenatal exercises on labor outcome among antenatal mothers. Here what we are doing, the manipulation is giving antenatal exercises. Okay. We have a experimental and control group. Outcome is what? The labor outcome we are assessing, that is the measuring variable. Okay. Now we will see another example here. Effect of post-operative lung exercises on post-operative lung, pre-operative lung exercises on post-operative lung expansion. Here, pre-operative patients are randomly assigned to experimental and control group. Experimental group will receive pre-operative lung exercises training. How they have to deep breathing exercises that they will be taught in the pre-operatively. Then the control group will receive the routine care which is there in that particular hospital. Post-test, that is lung expansion is recorded. Lung expansion is recorded after the surgery is over. So only outcome after therapy is completed. So this is the example. Now we will see another example, hand hygiene education program. One group of nurses receives training, other group will not receive. Compliance is checked after the training without pre-test. These are all the 3 examples. Now I will tell why we have to conduct without any pre-test, only the post-test, when the pre-test is not feasible. So I will give the explanation in the first. Yes, you go here. It is not feasible because labor outcome will happen after the delivery only. So here we are giving exercises in the antenatal period. Labor outcome cannot be checked in the antenatal period. That is why pre-test is not at all possible here. The outcome is what kind of labor they are getting if they undergo antenatal exercises. When we see another example, yeah, here the person who is admitted pre-operatively, for him you are giving lung exercises. It can be a deep breathing exercise, any lung expansion exercises you are giving. The effectiveness, lung problems you are assessing post-operatively. So pre-test is not possible because lung problem is normal. Lung is normal in the pre-operative period. In that situations, so when the pre-test is not feasible, means cannot be conducted, when outcome occurs only after the intervention. Yes, the labor outcome occurs after the intervention only. In these situations, after only design is useful. Avoids the pre-test influence, reduces the testing bias. When we go to the previous slide, let me go to the previous slide. So here, hand hygiene education program. One group, the compliance is checked after the training. Before itself, you are checking the hand hygiene things. Some people, the pre-test will have an influence on the post-test. What questions they have asked in the pre-test, they will remember, and that remembrance may have influence, that memory influence on the post-test. Without undergoing that program also, sometimes they may perform better. So the pre-test has an influence on the post-test. That is why this one has some remark. Yeah, here, avoids the pre-test influence, reduce the testing bias in such situations to prevent sensitization of the participant. What is sensitization? The participants will know what questions are there in the pre-test. Without properly undergoing the education program, there are the chances that they may perform better in the post-test. In these situations, we can go if the pre-test is not possible or outcome occurs only after the intervention, then it prevents the sensitization of the participants, or pre-test may influence the post-test. In such situations, we can go with the post-test only control group design. The second one is the pre-test post-test control group design. It is another name is classic experimental design, where the dependent variable is measured at 2 points in time, before the intervention and after the intervention. You see the figure here, random assignment of subjects to experimental and control group. Both the groups will have a pre-test, then experimental treatment is given, then the post-test will be conducted. This is called as a classic experimental design. Participants are randomly assigned, pre-test is given, intervention is given, post-test is given. So this design definitely helps to compare the changes over time between the groups. Now coming to the example, resilience training program for staff nurses. So in the pre-test, they will assess the stress levels are measured of the staff nurses. Experimental group will receive the resilience training program. Control group, no training. Post-test, stress levels again measured. This is a very simple example. differences in the stress reduction is evaluated, maybe from pre comparison of the experimental and control, post-test comparison between the experimental and control group, pre-post comparison of the experimental and control group. Many ways we can find out how effective this intervention program. Yes. Another example, hand hygiene training, pre-test skill competency, how well they are cleaning their hands. Then simulation based training is given, how to wash the hands, maybe a demonstration, such things. Control group will receive the just traditional lecture method. Post-test skill is assessed and the skill improvement is compared between the two interventions like a simulation-based training or traditional lecture method. In which method hand hygiene competency is improved will be assessed by this study. What are all the advantages? Yes, advantages, it measures the changes over time, control the baseline differences. For example, when we have a pretest, at the pretest, both the groups are equal. We can say that both the groups are equal. After the intervention, experimental group performed well. Justification can be done. So baseline, any differences are there, it can be controlled. Strong internal validity is there. It is more suitable for both clinical and educational studies. So one limitation is there. Sometimes pretest may have an influence on the post-test. For example, if it is a hand hygiene skills only, so they have assessed previously, the student may remember what mistake she has done. Then without intervention also, she may read the book and she may perform well. So that chances are there. Then the third one is the Solomon 4-group design. In the Solomon 4-group design, there are 4 groups are there. Easily you can remember, it is the combination of both post-test only control group design, pre-test, post-test control group design. So there are 4 groups are there. 1 experimental group and 1 control group are administered pre-test and other 2 groups are not. Post-test is conducted for all the 4 groups. You see here, there are the 4 groups. Experimental group 1 will have a pre-test. Experimental control group 1 have a pre-test. Then treatment is given, post-test is assessed. Experimental group 2 will not have the pre-test, only the treatment and post-test will be there. Control group 2 also only have the post-test. So 4 groups are there. We will see why the 4 groups. Example, effect of structured teaching program on knowledge of insulin self-administration. So here the nurse researcher wants to check if the structured teaching program improves the patient knowledge without any pre-test bias. 4 groups are there. The first group has a pre-test plus intervention. So group 1 conduct the pre-test. Knowledge is assessed in the pre-test. Intervention teaching program. What teaching program? Knowledge on insulin self-administration. How to administer injection, self-administration of insulin injection. Post-test is conducted again with the knowledge reassessed. Group 2, this is a pre-test only. Pre-test is conducted. No intervention is given. Again, knowledge is assessed. It is a control group 1. It is experimental group 1. So next, group 3, no pre-test, only intervention. Post-test is conducted. Group 4, no pre-test, no intervention, only post-test is conducted. So here you can see the 4 groups: pre-test, intervention, post-test. Pre-test, no intervention, post-test. This is experimental 1 and experimental group 1 and control group 1. These set is called as pre-test, post-test, control group design. The second one, experimental group 2, control group 2. This is called as after only design or post-test only control group design. So combination of these 2 designs is Solomon 4-group design. Why we need to do this one? When we do this one, what happened? Okay, so here in the previous slide, yes, one of the limitation of pre-test, post-test control group design is participants may have a testing effect if they undergo pre-test. That effect may be there on the post-test. To control that one, we are going with the 4 methods. Yes, we are going with the 4 methods, so that what happens, one group will assess the testing effect. Group 2 is kept for testing the effect. Okay, so in the sense, here pre-test we have no intervention, post-test. Only whether the testing has influenced on the post-test can be assessed without no intervention. If the control group 1, this is the group 2, is also improved, means there is a chance that pre-test is having influence on the post-test, to say that one. Then third group measures the pure intervention effect, because there is no intervention, no pre-test. Only intervention is there, then later knowledge is assessed. So here, how well the intervention has improved the knowledge. So the fourth group will act as a control, no pre-test, no intervention, only post-test. When we have a 4 group, we can say that pre-test doesn't have influence on the post-test. Only exclusively our intervention only improved the situation, to say that one. Next one is the crossover design. This is the 4th one. This is also called as repeated measures design. A crossover design is a research design where the same participants receive more than one intervention, but at a different time periods. Here only 2 things you have to remember. There is only receive more than one intervention. In the previous conditions, we have seen that only one intervention. It can be a knowledge improvement or training or otherwise self-administration, skill development, something else, only one intervention. Here the participants will receive 2 interventions at 2 different points. Instead of having a separate group, participants in crossover from one treatment to another treatment. We will see diagrammatically I have explained how it will be effective. I will tell you. Crossover design is equal to same participants, different treatments at different times. So this you remember. So here in this design, same subjects are exposed to more than one experimental treatment. Subjects are provided 2 treatments, one being experimental and the other control or reference. While one group receives the experimental treatment initially, the other group receives the reference treatment first. Okay, after a certain period of time, the treatment are crossed over, thereby allowing the subjects to serve as their own control. Diagrammatically, I will explain so that you will understand. Manisha, can you give feedback? Is it okay?
Yes, ma'am.
Okay. Because no, I cannot see audience, only my PPT is visible. That is why now and then I am asking the feedback. Okay, with the technical glitches, I cannot, if I departed, then it will be a problem. Okay?
No, ma'am. If anything happens, I will let you know.
Thank you.
No,
thank you.
ma'am.
Yes, here you see the, you just see the slide. Here I have given an example, a study to compare the effectiveness of massage and music therapy on the development of premature infants. So you all know that premature infants are having a less body weight. Weight is lesser than the normal children. Their development also very slow. So here we are giving 2 interventions. One is massage and another one is the music therapy. In this study, some infants are randomly assigned to first massage therapy, another group to the music therapy. Later, massage therapy newborns will come to music therapy. Music therapy newborns will go to the massage therapy. So here you see first pre-test group 1, group 2. First we are conducting pre-test. Pre-test means weight and development we are checking. After that, these group 1 people will go to the treatment A. For example, it may be a music therapy. After that, after music therapy, then we will assess their development. Maybe music therapy is given for a 1 month and see how much weight gain is there, what developmental changes have taken place. After some time, means they will give some gap. Maybe for 15 days, they will not give any intervention. After that, group 1 will receive the group B, that is the massage therapy. That massage therapy for a 1 month and again assessing the weight and the developmental changes. Then coming to the group 2. Group 2, this is another group. First, they will receive the massage therapy. Then their development will be assessed. Then the 15 days gap is given. Then they will go to the music therapy. So this is a thing. Now I will explain. Why this crossover? Crossover design is more useful when the person is having a chronic conditions, stable disease. Only a short-term intervention we want to give. See that which intervention is more effective. Whether the music is more effective or massage is more effective. Group 1 has received first music therapy. Group 2 has received massage therapy first. Yes, group 2 has received massage therapy first. So if, for example, group 1, their development is better, then you can conclude, compared to the group 2, then we can conclude that music therapy is better than the massage therapy. After some time, we are giving a gap without giving any treatment. Then again, that is called as a washout period. Means all the intervention should go out from the body. After that, this group 1 will go to the massage therapy. That massage therapy for 1 month, and again post-test, their weight and other things are checked. So, for example, compared to music therapy, if the massage therapy is effective, means these children's development will be fast during that 1 month period. So, like that, they have a 2 groups. One group will have a 2 intervention. Another group also 2 interventions, but one group will receive 1 treatment first. Another group will receive another treatment first. So, for example, these knee joint pain. So, there are 2 medications are there. We want to know which medication is effective. So, if it is a previous thing, only 1 intervention we can test. With crossover design, we can test whether which medicine is better for them. For example, group 1 knee joint pain people will receive one kind of, for example, treatment A. For 1 month, they will take that medicine and they will ask you how much pain reduction. After that 15 days, they will not give any medicine. So that whatever is there in their body, that medicine has to come out. After the washout period, then again 1 more medicine they will give for a month, and again they will assess the pain. They will ask the patient, when you have taken a treatment A, you feel better, or treatment B, if you have taken, which is more better. Same like that, group 2, what they will do? The treatment B they will receive first, then treatment A. By doing the crossover, they will become their own control and they know which treatment is working for better. With this, we can compare which intervention is better, which treatment is better than the other one. Yes, what are all the advantages? Requires the fewer participants. When we taken the Solomon 4 group, you need to have a 4 groups. Here it is not required. Reduces the variability. Each participant will act as a own control because 2 interventions it is receiving. 1 intervention first and the second intervention, so that they will act as their own control. More statistical power compared to other designs. But limitations, it is not suitable for permanent treatment. It is only a short-term treatment. Okay, because no permanent treatment means it will be there in the body. So this treatment will have effect on the other treatment. So washout period is given to remove that treatment effect. Carry over effect possible. However, 15 days you have, 1 month you have given a time to get rid of that treatment. Sometimes the treatment effect will be there in the body. Needed proper washout period. For each drug, you should know the lifespan and how many days it will be there. For example, if you are giving a music therapy or massage therapy, 1 month you have given, the effect will be there how many months in the body. Based on that, washout period need to be fixed. Not suitable for life-threatening conditions. Then factorial design. So we have completed post-test only control group design, pre-test post-test control group design, Solomon 4 group design, crossover design. 6th one is the factorial design. It is an experimental design in which 2 or more independent variables are studied independently. It helps the researcher examine individual effect of each factor, combined effect. So with the example, I will explain this one. A study to evaluate the effectiveness of relaxation and distraction. 2 interventions on reduction of combining 1 intervention with the another on reduction of pain among cancer patients in 2x2 factorial design with 4 cells are used. Each cell contain equal number of subjects. Cell A subjects receive distraction and relaxation intervention. Cell B subjects receive only distraction. Cell C subjects receive only relaxation. Cell D subjects receive no intervention and serve as a control group. Yes, you see here. So we divided into 4 groups, like a Solomon only, like that here 4 groups. But group A, for example, 25, 20, so group A, 50 sample, group B, 50, group C, 50, and group D. Total 200 sample, 4 groups we have made. Group A received both distraction and relaxation therapy. How much the pain is reduced? So group B have received only distraction, no relaxation is given. So group C, no distraction, only relaxation. Group D, no intervention at all, it is act as a control group. What is the benefit having like that? So with this, we will come to know combined interventions are better than individual interventions. At a time, 3 hypotheses we can test. Whether distraction and relaxation more effective in reducing the pain, only distraction is better, only relaxation is better. Without anything also pain is decreased. So with this, no, we can test 3 hypotheses at a time. So more useful in studies, multiple interventions we can test, whether combined is okay or in a different interventions are okay. Without intervention also the patient is improved. Identifies interaction effect, it saves the time and resources, high research efficacy. Then what are all the advantages of factorial design? Test more than one variable, as we have seen that distraction and distraction and relaxation, both we have checked. Detect the combined effect by adding together, is there any benefit, or alone is there any benefit? It is economical, strong analytical value. Limitations are complex data analysis, because 4 groups are there, analysis will be little difficult, larger sample size is needed. Then interaction, interpretation may be very difficult in this condition. The last 6th one is the randomized block design. A randomized block design as an experimental design used when the researcher wants to control the effect of known variable. So called as a blocking variable that might influence the outcome. Participants are first grouped into homogeneous, then randomly assigned to the treatment within each block. This also, by reading this one, you may not understand. With the example, I will tell. Steps are, subjects are divided into homogeneous groups. For example, we have a sample, males and females are there. Block A is male and block B, like a females, we will make it. Then subjects within each block are randomly assigned to experimental and control group. For example, in the males only, we have a experimental group and control group. Again, in the females, we have a experimental group and control group. Then subjects in each block are pre-tested. Block A and block B experimental group subjects will receive only the experimental treatment. Block A and block B control group subjects do not receive any experimental treatment and act as a control group. Subjects in all the group are post-tested. You see here, one example here. So here we have a 4 groups, male and female. Again in the male, we have a control group and experimental group. Again in the female, we have a control and the experimental group. We want to check the vaccine and placebo effect on this male and female. The researcher makes 2 blocks based on the gender. Within each block, subjects are assigned to experimental and control group. So here, one group will receive the vaccine, experimental group male and experimental group females. Then one group, control groups will receive, male also control group is there, female also control group is there. They will receive the placebo therapy. So this is the thing, the student, then they will compare the results. With that, we will come to know whether the men is having more efficacy on the vaccination or placebo therapy, women is more efficacy on the placebo or vaccination therapy. So here we are constanting the gender, that too, we are making a groups like a male group separate and a female group separate. That is called as randomized block design. Pain management study, research question. So we can also have many groups. Effect of music therapy on post-operative pain. So here we are making groups like younger adult, middle adult, older adults. 3 groups we have made. Again in the 3 groups we have experimental and control. Total 6 groups it will become. 2 into 3. So then for each group you have a music therapy and control group. And middle aged also music therapy and routine treatment. Old age also music therapy and routine, so that we will know which age group people are having more effect on the music therapy compared to others. Why nursing researchers use this blocking? It controls the known confounding variables. We know that teenagers are very much means influenced by the music. Such times we can control having a teenagers only separate, middle age only, elderly people. It improves the more precision, because we can say that these females are more influenced, or efficacy rates are high in the females when we give vaccination or placebo, reduces the experimental error, enhances the internal validity. So randomized block design, the advantages: reduces the variability, more accurate comparison because we made it a subgroups. Useful when the sample is heterogeneous, sample is a mixed of many things are there. This study means this kind of studies are useful. Strong control over the bias. Limitations: need correct blocking variable. Complex design requires a larger sample because 4 group teams are there, or otherwise 2 into 3 means again 6 groups are required. Not possible if the blocking variable is irrelevant. Yes. Now, strength and limitations of the true experimental design. The first strength is more scientific in nature. All 6 things are more scientific in nature, but one limitation: artificial in nature. Most powerful method compared to quasi-experimental designs, but due to ethical reason, certain variables cannot be manipulated. Highest level of evidence-based practice is there in the true experimental designs. Pre-test can affect the post-test response. These are all the strength and limitations of true experimental design. Let me review. There are 6 types are there in the true experimental design. In the true experimental design, the main important thing is manipulation, control, and randomization. Subjects should be randomly assigned to the group. There should be a control and experimental group. Manipulation means addition or modification or deletion of the intervention should be there. When these 3 components are there only, true experimental design, it will become the 6 the types are there. Post-test only control group, pre-test post-test control group, then crossover design, Solomon 4 group design. Okay.
Ma'am, are you there? Ma'am, are you there? I think she's having some technical issue. Let me check it. Call. Yes, ma'am. She's faced some technical issues. Just I had connection with ma'am. Just she's sharing the PPT again, and she will continue the session. Just have patience. So by the time ma'am is getting connected, so we can interact with each others. I think many of the people they are most of the students and I think colleagues they are from the nursing background only.
Yeah.
And we
I
get
think I have connected now. Is it okay?
yes, ma'am. Yes, ma'am. It's okay.
Okay. I'll share now.
Yeah, sure, ma'am.
Is it this one, you know? Share. Is it okay?
Yes, ma'am.
My slides are visible?
Yes, ma'am. Your slides are visible.
Okay, everything is fine. Shall I go ahead then?
Yes, ma'am. You can continue.
Okay. The next method is quasi-experimental designs. Quasi-experimental designs are called controlled trials without randomization. So here I told that three things. No, one is randomization, control, and manipulation. Manipulation is must in the experimental designs. So here experimental and control groups are there, but not randomly assigned. Maybe some reasons like a ethical consideration, difficult for randomizing the subjects, difficult to randomize by locations, small available sample size when only very limited sample is there. Again, randomization, it may not be possible in such situations. Quasi-experimental designs, there are only four types are there: non-equivalent, control group, pre-test, post-test design. After only non-equivalent control group, one group pre-test, post-test design, and time series design. Four methods are there. In the previous true experimental design, I explained about the 6 designs. Now we have a 4 designs. So the first one is the non-equivalent control group design. It is a quasi-experimental design where there is an experimental group and control group is there, but no randomization. Pre-test and post-test is conducted and intervention is given. When the random assignment is not feasible, this design can be structured teaching program on breast self-examination. Here the researcher studies the effectiveness of teaching program. What he did, women from community A, he has taken for the experimental group. Community B, he has taken for the control group, because to prevent the intervention contamination in the same community, if you have taken experimental and control group, what happens? There are the chances that these people will share the information to others. Invariably, control group people also sometimes they may do better. For that reason, to avoid the contamination, exclusively one community woman is taken for the experimental group, another community has taken for the control group. So pre-test knowledge is assessed for the both the groups. Teaching is given only to the experimental group. Post-test knowledge is reassessed. Groups are naturally exist and no randomization is done here. One used in research. Sometimes randomization is impractical. For example, in one ward only we want to do for one patient changing the position. In some patients it is not changing, it is unethical. Works with naturally existing groups, suitable for both educational and clinical studies.
Ma'am, are you there? Ma'am, are you there? Yes, ma'am, your slides are not moving, ma'am.
Not moving.
Again, ma'am, she is facing some technical issue. Just wait for a while.
So which slide I have to restart? Any idea?
47.
Now is it moving, my slides?
Yes, ma'am.
Okay, no?
Yes, ma'am.
Okay, okay. So can I start from the types of quasi-experimental design?
Yes, ma'am, you can.
Okay. So there are four types of quasi-experimental designs: non-equivalent control group, pretest, post-test design. After only non-equivalent control group, one group pretest, post-test design, and time series. Non-equivalent control group, I already explained that there is an experimental and control group, but groups are not no randomization, pretest and post-test. Where the randomization is not feasible, then we'll go for this kind of designs. So structured teaching program on breast self-examination. Researchers studies the effectiveness of teaching program. Experimental group, women from the community A. Control group, women from the community B. Pretest knowledge is assessed in both the groups. Intervention is given only for the experimental group. Post-test is conducted both the groups. Here, why we have taken this design? This design means because the contamination may happen. In the community A itself, you have an experimental and control group. What happened? When you, for one group, you have given a breast self-examination, they will share the information to another group. So that invariably control group also improve in the assessment. To avoid that one, to avoid intervention contamination, groups are made in a different communities. Then randomization is often impractical, works with the naturally existing groups suitable for educational and clinical studies. Major limitations, selection bias are there. Maybe a group, community, which you have selected, they may be all educated and having already high knowledge, and they are very cooperative, then results may come wrong. Yes. The second one is after only non-equivalent control group design. It is another thing like a post-test only control group design is there, no equivalent to that one, but no randomization. It is a quasi-experimental design where the two existing groups are used, no randomization, no pretest, only outcome is assessed after intervention. Experimental group, pressure ulcer prevention strategy. Experimental group, ICU using the new protocol. Control group, ICU using the routine care. Outcome, pressure ulcer incidence is observed after giving the protocol, after implementation of protocol. Only post-intervention comparison, only one-time comparison is there. Why this design is used? I already told that where the pretest is not feasible, avoid the testing effect, practical in clinical setting. So major limitation, selection bias because no randomization. Since no pretest baseline equivalency is not known, whether both the groups are equal or not, we do not know. Is it okay, Manisha? Am I there?
Yes, ma'am, you are there.
Okay. So one group pretest, post-test design. Most of the books it is given like a pre-experimental design. Only one group is studied. Outcome measured before the intervention. Intervention is given. Outcome is measured after intervention. There is no control group. Only one group is there. No randomization. No control group. So here at one assessment is given. Intervention is given. Post-test is given. That's all. Stress management program for the student. Stress levels are measured. Intervention, stress reduction intervention is given. Stress is measured again. Why it is used? When the very simple to conduct, useful for pilot studies. When you have a resources are less and only one group is available. Suitable when control group is not feasible. Good for educational evaluations. Major limitations: no comparison group, weak internal validity. The first one we have completed. Non-equivalent pre-test post-test control group design. One group pre-test post-test control group design. After only design. Non-equivalent post-test only control group design. 3 is over. Fourth one is the time series design. It is a quasi-experimental design. Outcome is measured multiple times before and after an intervention. It helps to detect the trends and the intervention effect, useful when randomization is not possible. So here we will see pre-intervention, the pain management protocol. Some pain, new treatment you are giving for the pain. So pain, no, especially when you take a back pain or knee joint pain or body pains, not constant. Usually when we go to the doctor, last 1 week I am having a problem. So one day it may be severe pain, another day it may not be there. So it will be fluctuating, especially mood also. One day we may have stable mood, stable pain will not be there. In that conditions, pain scores are recorded daily for a 1 week. So to Monday pain, Tuesday pain, Wednesday, Thursday, Friday, Saturday pain scores. After 1 week, one number, average number they will take. Then they will give the new treatment. Okay. Then after that, the new treatment is given for a month. After that, 1 week again pain is assessed. So new pain management guideline, post pain scores are recorded daily. It identifies the sustained effect because after immediately taking 1 or 2 days, you may feel better. Again, pain may come. So for that, repeatedly, they will measure the pain. So this is called as repeated measures. Why time series is valuable in nursing. Controls the history effect better than the simple designs. Detect the trend over time. What changes have happened? Repeatedly assessing the pain. Is there any improvement or decreased? Show immediate versus sustained effect. Sustained. So the pain reduction is maintained for a month or for a 1 week, the pain reduction is there. So to say that one, it is more practical in the hospital settings. More observations is equal to better internal validity. So time series decision stronger than one group design, identifies the pattern, useful for policy intervention evaluation. Limitations: no true control group. External events may influence the outcome, requires the repeated data collection. So then I have completed, there are 10 types are there. So 6 types in the true experimental design, 4 types in the quasi-experimental design. So let me go back to the quasi-experimental design. Yes, there are 4 designs: non-equivalent control group, pretest, post-test design. After only non-equivalent control group, one group pretest, post-test design and time series design. Manisha, connection is there? Again, go on.
Connection is there, ma'am. Don't worry.
Okay, okay.
Share the slides again, ma'am. Share the slides again. We are not able to see the slides.
Okay, okay.
Yes, ma'am.
So now is it okay? Maybe stop sharing might have happened. Okay,
Yes,
then.
ma'am. You just check it once.
Is it okay now?
No, ma'am.
Is it okay now?
No. Yes, now it's okay, ma'am.
Okay. Now I have completed actually. Just to let me review whether you understood or not. The first question is, in a post-test only control group design, the major threat minimized is. Audience can answer, Manisha, or that scope is there?
Yes, ma'am.
Yes. Which is the correct answer? History effect, testing effect, maturation effect, and instrumental bias. Which will be minimized?
All the audience, kindly just unmute yourself and try to answer
Yeah,
the
please
questions.
try to answer.
Yes, try to answer the questions.
Whether they are there or went for the lunch break, we don't know.
No, ma'am, they are there.
Okay.
They are trying their level best for doing things.
Question again, ma'am. Yeah. In a post-test only control group design, the major threat minimized is A, B, C, D is there. Which is the correct answer?
Miss B, madam.
B is the right answer. Very good. Testing effect can be done with the post-test only control group. The Solomon 4 group design primarily controls for selection bias, carryover effect, pretest sensitization, or attrition bias. Connection is there?
The connection is there, ma'am.
Very good. Pretest sensitization is the right answer. In a crossover design, each participant serves as.
Audience, try to open, just be on camera, just answer the questions. Unmute yourself and just on your cameras and then answer.
A, B, C, D.
It's a random
Very
sample.
good. Their own control. B is the right answer. Which design is most appropriate when studying the interaction effect of interventions? 2 interventions at a time we can check.
Factorial design.
Very good. Factorial design is the right answer. Blocking in a randomized block design is done to control what? Little higher level these questions. I have not explained in my presentation. Yes, you can answer. All these questions.
Variable B.
Very good. Very good. Extraneous variables, known extraneous variables can be controlled by randomized. What is the limitation of crossover design? I told about washout period.
See carryover.
Huh?
See carryover.
Carryover effect is carryover effect is the limitation because it may carry, another intervention may carry. Yes. Coming to the summary, I explained about the definitions of nursing research, then basic types, experimental and non-experimental. Under the non-experimental, we have a descriptive and correlational designs. Under the experimental, we have a true experimental and quasi-experimental. Advantages of all 10 types which are commonly used in the nursing studies. Limitations also I have explained. So this is the textbook I have used for my reference. And this is meant for 7th semester students. Now it is available in the market and newly released. These are all other my textbooks you all may be knowing. Mental health and psychiatric nursing, applied psychology, applied sociology, nursing education, practical record book. Now I made a behavioral sciences separately for the GNM. It includes both psychology and sociology. Separately for GNM students, mental health and psychiatric nursing book is available and health informatics. So you have any further doubt or anything, you can contact me over this email ID.
Thank you for this wonderful session, ma'am. Because we